What's up, Hippie Bio Penguins? So, hi, we are coming to you live from beautiful Georgia. Yeah, it's beautiful out here today. So we're going to do a CRAM session today. So if you haven't been to any of my other sessions, you know that I talk fast. And so today is going to be nothing different than any normal session where I speed run through basically as much as I can get through. So this will probably last longer than an hour just because I like to talk and tell you as much as I can tell you. And it will be recorded. So if you're wondering about, oh, will this be recorded? It will be recorded. It will be posted on my website as well as it will be posted here on YouTube. And then I'll be sharing it out on my Instagram. So, hey, I'm Mrs. Jones. I'm AP Bio Penguins. So when we started, I said, what's up, AP Bio Penguins? Why am I calling all penguins? Well, because of the fact that you were in this session, you are a penguin and you are dressed to success. So in 5 days, or whenever you're watching this recording, you're going to take that AP Bio exam. And no matter what happens on that exam, you're going to do your best. And your best is always enough. And because you're a penguin, you are ready and you are going to rock this thing. Okay. So today's format, we're just going to first talk about the exam. What does it look like? I'm going to do a quick overview of the units. If you're needing more information about any of the units, you're welcome to go to any of the other YouTube videos. I've got YouTube videos for every single unit. I've got videos for every single topic in the CED that I made them into TikToks. I've got games. I've got so much stuff for you. So if you're needing a little bit more detailed information, you could go to any of those other ones. We're going to do some practice questions that are kind of intermingled in there, as well as we'll have Q&A. Okay. So I've got Miss McClinton in the chat. She's going to moderate for us. And so you're welcome to write whatever you want in the chat. I noticed that there's kind of a delay. So she's in there to try to help me to answer some of the questions that y'all might be having, as well as I've noticed in other sessions that y'all like to help each other. So if you know an answer to something that somebody's asked in the chat, go ahead and answer them back. And at the end, we'll just do some quick Q&A or whatever we need to make sure that we are all ready and prepared for this exam. So without further ado, let's get started. So first thing, there are 2 sections to the exam. The first section is going to be 90 minutes and you have 60 multiple choice questions, and this is worth 50% of your exam. And that's your first section. Then you'll have like a 10-minute break, I think, and then you'll go into section 2, which will be your free responses. In free responses, we've got 90 minutes, and then we've got, there's 6 questions, there's 2 long ones and 4 short ones, and this is also worth 50% of your exam. And so half of the score comes from multiple choice, half of them come from free response. So for my students, I tell them that they probably want to have most of their points coming from that multiple choice. Like you tend to be able to get more points in multiple choice. So students always ask, like, what do I need to have in order to get a 5, 4, 3, 2, 1? I'm not going to play that game, but I am going to play the game of, okay, if we look at the 2020 scoring guidelines, you need a 54 out of 120. Now, I'm not very good at quick math, but I do know that that is less than 50%. It's probably like 45%. That you need to get on the exam. Now, the exam scoring guidelines were kind of realigned a couple years ago, and actually that number is a little bit lower, but I don't trust that number completely. So I wanted to give you a number that I trusted, which was the 54 out of 120 will get you a passing score on the AP Bio exam. And I think that we can get at least 50% of the points. I think so. And so that is what your goal is, to aim for that, but I always want you to go aim a little higher. So my students always tell them I want you to aim for the 4, so you could be surprised by a 5 and fall to a 3. So when I say fall to a 3, I mean, like, if you just happen to not know something, if you were having a bad day, like sometimes you don't get the score you were hoping for. So if you're aiming for that 4 and you happen to miss your mark, you still are going to get a 3, which will pass the exam. So there's 8 different units. I'm assuming at this point you know this. And so the first unit is on chemistry of life. And so according to the CED, they tell us approximately what percent the exam should be in each thing. And so it should be 8 to 11%, which would be about 5 to 7 questions. And so I analyzed the 2020 exam, and that was 5.7 questions. And then if I pulled up the 2022 and 2023 kind of exams that were released to the teachers, that was 6 questions on that. So like that's a good number right in the middle. There's probably about 6 questions on unit 1. Unit 2 is on cell structure and function, 10 to 13%, so 6 to 8 questions. And it did fall within that mark of about a little more than 6, closer to 7 kind of for those questions. Unit 3 is one of our bigger ones. It's 12 to 18%. So it's about 9.3. So somewhere in that ballpark of 9 questions is where we saw unit 3. 4 is on cell communication and the cell cycle. No, 90 minutes for 60 multiple choice is correct. You have about a minute and a half per each question. Um, and then for self-communication, that was 10 to 15%, 6 to 9 questions. It did fall on the lower end on the most recent exams. So it's probably more in like this kind of that maybe like 6.2 ballpark of life. Then Unit 5 was on heredity. So 8 to 11%. This was about 6 questions, and there was like kind of more in the 7 ballpark for, yeah, cellular respiration is part of the curriculum. So that was in the kind of the 7 ballpark recently. Unit 6 was on gene expression, so another larger. This one out. So 12 to 16%. So about 8 questions there. Hopefully the audio still is good without my earbud in. If not, I can switch to the other earbud and try that one out. So natural selection was on 13 to 12%, so another 20%. So that's another one of the bigger chunks, 9.3 questions in the 2020 exam. And then Unit 8 is going to be ecology, which for the most part, either A, your teacher may have given that to you as a summer assignment, or B, they quickly ran through it as they were kind of finishing things out. So that's kind of like what the different topics that we're looking at are, okay? So the first section we said is multiple choice. So there's different types of questions we're going to have. We'll have independent questions, which means that we've got a small prompt, and there's maybe like a graphic, or maybe like one picture, and you're analyzing that one picture. There may be like a chart or a graph, but there's just going to be a very small little bit of information you're going to have to internalize. And then there'll be one question. Now your set questions are going to be kind of these longer prompts. There'll be this big, long readings passage. There'll be kind of multiple graphs, multiple charts. There's a lot of information to kind of analyze and bring in. And this is usually about 3 to 5 questions. It could be up to 6, but it's, minimum would be at least 3. Okay. So if you look at the 2020 exam, there was 31 to 38 independent questions and 22 to 29 set questions. I didn't think to analyze the 2022 and 2023 exam until literally 5 minutes before my stream started. And that's when I just quickly wrote down the unit numbers. But so it's probably going to fall in that same ballpark where you've got a couple more of the independent questions and less of the set questions, because they probably take a little longer to get through those set questions. So this is what an independent question looks like. You're going to have, again, a short little prompt, and then you've got some questions for it, versus your set question. I know I flew through that fast. Is going to kind of give you a graphic, and then there's a prompt. Kind of in the more recent exams, we've seen kind of multiple graphs in which to kind of help out with that. And then this one has 5 questions that correspond with that. Okay. So in terms of timing, how long should you spend on each one of these? Okay. So you have about a minute and a half per question. But I find that some questions, you can get through them really quick. And some questions, you sit there and think a little longer. You have to read the prompt. There's a lot more that goes into it. Okay. And so I tell my students that for every 10 questions, look at the clock. Make sure that only 15 minutes has elapsed. If less than 15 minutes has elapsed, you're doing good. Keep rolling how you're rolling, okay? If more than 15 minutes has elapsed, then you probably want to pick it up your pace a little bit, okay? Or you need to know yourself and know, okay, well, I'm going to put in the extra time to make sure I get these questions right. So maybe the later questions, I'm going to have to kind of Christmas tree them, but I'm going to focus on these and make sure that these questions are right. You can decide that for yourself, but I just kind of wanted to give you a ballpark to figure it out. So 10 questions, check the clock for 15 minutes, because again, you have 90 minutes for this. Okay. So when you're going through them, you want to annotate your questions. So if there's any important words, you want to kind of underline them as you read. Make quick reference notes to prompts and figures. So if you notice that there's an increase and a decrease, like you see some type of correlation to it, go ahead and kind of jot those notes so that when you come back to it, when you're doing those set questions, and you're like, oh, I need to figure out what the relationship is. Oh, I can see the relationship. And you already have that answer. You've already analyzed it as you were reading that prompt. You came up with that information. And write on your graphs. Show your work for the math. Y'all, there's math in here, and we both know that you are going to make a quick small error if you were just typing things into your big TI calculator. You forget the parentheses, you forget some unit, something somewhere, okay? So go ahead and show your work and kind of jot down what you're doing so that you can make sure that you don't make those errors, okay? Trust yourself. So cover up some of the answer choices. Maybe like you sometimes know the answers without looking at the answers. And so if you come up with the answer on your own, then there's a higher chance that that is the right answer. And then use your resources, use those figures, use those diagrams to help you. There's a lot of science practices in this thing. And so they're determining, can you read a graph?
Can you analyze this diagram? Can you bring some stuff together? And so use those figures, use your resources to make sure that you can answer it. I tell my students all the time, like, dude, all you had to do was say that this thing was cut off. Like, the picture shows it there, and then the picture shows it not there. Like, it got cut off. Like, it's super easy to follow that, okay? So then for section 2 is your free response. We're going to have 2 different types. You've got long FRQs. There's 2 of them. The first one is interpreting, evaluating experimental results. They're going to give you some data from some experiment. They may ask you some questions about that experiment, okay? And the second one is going to be your graphing question. Yes, you are going to have to graph. The second question on your test is going to be a graphing question, okay? So make sure you scale those axes. Make sure you label them. If you're running short on time, get at least the labeling done. You can get a point for labeling your axes. And recently, you can get a point for just saying what kind of graph it is. So if you can't get the whole graph made and you know it's a bar graph, just draw a couple of the bars so that your readers know, oh, they know that this was a bar graph, and we can go ahead and give them that point. Like, try to get the points wherever you can. Of course, I think you should do the whole graph because the graph is worth 3 points. But if we can get anything because you're running out of time, get something on paper by getting at least your axes scaled, getting your axes labeled, and maybe showing them what kind of graph it's going to be. Short FRQ, these are going to be worth 4 points each. Oh, I forgot to mention. So for the long FRQ, it's going to total 18 points. And so like if your first question is 8 points, your second one will be 10. If the first one's 9, it'll be 9. And then if it's 10, then the second one will be 8. So it's always going to total 18 points there. And you can usually tell the points based on the bolded terms. They did change the exam this year in which they're trying to kind of break up that reading a little bit. So you're probably going to see a little bit of reading, and then you'll see the first question. And then you'll see maybe your graph or your data table, and then you'll see your next question, okay? So they are breaking it up a little bit for you. So for those short FRQs, they're 4 points each, and you're going to see A through D. Like, they are 4 broken apart. The first one is scientific investigation. Again, you're going to use those scientific evidence. You're going to use your experimental design for those. Second one is conceptual analysis. Do you know biology? The fifth one is analyzing a model or visual representation. So that means you're going to have to annotate in some way. So in the 2021 exam, you were drawing what was going to happen in late July for this field where there was an invasive species. In 2022, you were, you were drawing an invasive species on a food web. And then for 2023, you were just putting a mark on a phylogenetic tree. And we've got that later on in this activity today, okay? And then Unit 6 was on analyzing data. Last year, it was a box and whisker. So make sure you do know how to work a box and whisker. We're going to look at some of that, kind of how to read a box and whisker later on today. But do make sure you know box and whiskers, okay? Now, there are some questions coming in the chat about what you can use. So for your graph, I recommend you use a pencil. You can do the graph in pencil, and if you have time, go back at it in pen. Calculus, stats, I think maybe one of the physics, they use pencil for their entire exam and their exam scores, okay? So I would use a pencil for your graph and then go back over it with pen. Do not use a erasable pen anywhere on your test. A erasable pen is going to be erased by heat. These things, these tests are going to be packaged up into boxes. They're then going to be taken in trucks, hot trucks. It is hot outside, to go to a warehouse, and then they have to be scanned in. There's a chance that your erasable pen could be erased. Do not use a erasable pen. Do not use the pen that bleeds. Make sure that you are using like the basic Bic, okay? Like super simple, easy ones, okay? Hey, Marjan from Marco Learning. We'll see Marco Learning on, what day is it? Wednesday. We'll do this whole thing again on Wednesday with Marco Learning, by the way. Okay. So 2, we're going to be graphing, and 5, we're going to do modification of a diagram. That's something that's going to be in your book to kind of help you to kind of know that you're going to have to make those specific things in the book. Okay. There are these certain task verbs that you're going to see in it. This is kind of a screenshot that shows all the different task verbs that they have. So making sure you know what these task verbs mean will help you to ensure that you are appropriately answering the questions that they're asking. Okay. So strategies. Okay. So let's talk about timing first. Take about 5 minutes and read through your questions and renumber them. Okay. If you know the second question really well, you can answer that second question first. And then let's say you know question 5 and you know question 6 really well. Go ahead and answer those, and then come back and do question 1, and then 3, and then 4. Do them in whatever order you know them. This is a points game. Our goal is to get as many points as possible, okay? So read through, figure out any of them, make any kind of quick notes that you need to, okay? And then you're going to give yourself 1 long, 2 shorts, and then 1 long and 2 shorts. For the longs, you'll give your 12 20 minutes. For the shorts, you'll do 8 minutes. Strategies I've seen some students do is that they write the time at the top of the page. And what that means is that at that time, they move on. Even if they're not done with that question, they move on. And that's a way they can actually answer every single question and at least get the things that they know on the paper. Okay. So if you give yourself 20 for the longs and 8 for the shorts, that gives you about 10 minutes. It's actually more like 13 minutes, but you know, things don't fit perfectly to time. So you have about 10 minutes to read back over your responses and add any extra information. If you had to move on from a question early because you were still thinking things through, go back to the question, answer those questions, make sure that you have as much as you can, okay? So format, read the question. I don't think you heard me. Read the question. Oh yeah, and read the question. So sometimes I find that students are answering the question and all of a sudden they forget what the question is asking and they go back and they're like, oh, I'm not answering the right thing, okay? So make sure you're answering what they're actually asking. There's a mnemonic device that some teachers use. It's called ATP. Answer the prompt. Answer whatever they're asking you, okay? Be sure to label your responses. You don't have to label your responses, but it does make it easier for the readers. So I recommend labeling your responses. And then you want to write in knowledge order. If you know C, go ahead and answer C. Answer C first, and then go back to D, then go to B, then go to A. It doesn't matter what order you answer them, just answer them. So that means at the top of your page, you'll have D, then you'll have B, then you'll have A, then you'll have C. Like it does not have to be chronological order going down your page. Just label your responses so we can easily find that. I can use the other earbud because that one went out. So I don't know whether I can get this one to work, but I can try to do that. I'll get this one charging in the meanwhile, and hopefully it will connect and we can be good to go. So you want to write in knowledge order, okay? So in the order that you know the information is how you want to be answering them, okay? You also want to make sure that, sorry, I'm going between the screens to try to get the earbud to turn on, and now I got lost where I was. Okay, so logistics, you want to be aware of any type of contradiction, okay? So contradictions mean that you say one thing and then you say something else. So like the mitochondria is where we're going to see oxidative phosphorylation. There'll be the most amount of ATP produced. And then 2 sentences later, you say, which is why we saw the most ATP produced in the Krebs cycle. Be very careful that you don't kind of go back on yourself and say the wrong thing, okay? So yeah. I'm still trying to find out where my earbud is. It has gone missing in terms of the like connecting. So hopefully you can still hear me and that I did not completely lose everything. Okay, so where are we? Because I need to pull the PowerPoint back up. I don't like doing this in lives. Okay, so helpful resources that we have, okay? So what we could be doing, okay, is of course AP Bio Penguins. If you are not on AP Bio Penguins, you need
you what I have over here, okay? So if you pull this drop down, you're going to see there's all of my review sessions here. I've got the 10 days AP. I've gone through and I've kind of given you a plan. I know we only have 5 days left, but I'm just giving you some more information there, okay? FRQ Fridays, I've gone through and I have done an FRQ Friday video for every single FRQ in the CED. On AP Central. Go through, pick any random question you want, and you can pause the video and try to answer the question. And then I, of course, explain it. And then I give you a student answer. I show you them, okay? I've got 120 quizzes, games. I've got TikTok reviews, okay? There's literally a TikTok for every single standard in the CED. So if there's a certain topic that you're struggling with, go ahead and answer that one, okay? Or go watch those videos. I've got all my macro learning sessions on here. I've got all these Pure Penguin things. I've got so many resources on here that you could just get lost in there, okay? The volume's better. Awesome. I'm glad the volume's better. I thought this earbud didn't work, and that's why I wasn't using it. I never use this earbud, okay? So there is the website to kind of help you out, okay? So podcasts, okay? You're a podcast person. You want to listen to something as you kind of walk your dog, walk your fish, walk whatever it is you walk. Absolute recap, okay? So Melanie Kinga is amazing, okay? So she's got a podcast for every single topic, okay? As well as she's also doing videos. She's going to be my competition on Wednesday when I go live with Macro Learning. She's also doing a live stream, so you can check her out. She's got amazing resources. Also thinking about like YouTube, we've got Bozeman Bio, we've got Crash Course Amoeba Sisters. My students love this guy named Gabe Poser, okay? So is it sponsored by AP Bio Penguins? I am AP Bio Penguins. So my students love Gabe Poser. So there was somebody earlier today on, oh gosh, what was I on? Discord. and they were bad talking AP Bio penguins and saying that I talk too much, which is fine, whatever. So I told them, go check out Gabe Poser, go check out Carrera. Those are the videos that my students actually enjoy watching, those people. So go watch them if I'm not helpful for you, okay? You could also go and get a review book. At this point, we're at 5 days. You want to go to the library and borrow the book so that you don't have to buy it, number one, and number two, like it's so short of amount of time, you're going to be kind of spot checking, getting the topics that you need a little bit more information on, okay? Practice questions. AP Classroom has tons of them, okay? So get your teacher to open up the progress performance checks, get everything like that you can on AP Classroom, okay? AP Central has a ton of free responses. They've got the CED. In the CED, you've got practice multiple choice and free response questions in there, like. And I even did a video for those FRQs too, okay? No, there's no Discord for AP Bio Penguins. I'm in the AP like Discord that like somebody made. It's like the AP students Discord. And then there, if you want like study cards, there's this company called Etch, it's like learnetch.com. If you go to them, they'll have short answer questions, and then they give you explanations after it. You can, of course, use Quizlet and Quizzes, but the great thing about the Etch is the fact that they've got explanations for everything, plus they link you to my videos. And then, of course, all kinds of other stuff there. And they have it for AP US History, and they link you to Heimler's videos, okay? So let's practice, okay? So how can we practice? We can look at the 2013 release exam. That's the only exam that they have open for you that you can access, okay? AP Classroom is another place where you can get a whole bunch of resources, okay? Free response, if you want to practice your free response, AP Central. They've got all the way from 1999 to 2023, minus 2020. And they've got the questions, they've got the scoring guidelines, they've got student responses. That's all there for you to help you to review, okay? Whew, now that we spent 20 minutes going over tips and tricks, let's talk about the content. And as you can tell, there's no way I'm finishing this in an hour, but I'm going to keep on going. And whenever I finish, I finish. Miss Clinton, whenever you need to leave, you're welcome to leave. So first thing, hydrogen bonds. We need to know hydrogen bonds, okay? So these are just going to be attractive bonds, okay? There's no actual physical connection between them. They're attractive, okay? So they're very weak bonds. And they're between a oxygen that's bonded to a hydrogen that's attracted to another electronegative atom, okay? So the partial positive that's on the hydrogen is attracted to the partial negative that's on that electronegative atom, like the oxygens, the nitrogens, and so on, okay? And so there's like this kind of weak bond between them, okay? You're gonna find those hydrogen bonds in your proteins. You'll find them in your nucleic acids, okay? So it's important to know where those are gonna be. So we'll talk about that in a second, I promise. Okay. You also kind of think about different types of bonds. So we've got covalent bonds, we've got ionic bonds. Covalent bonds are going to be sharing valence electrons, while ionic bonds are going to be attractive. You got a partial positive, you got a positive and a negative due to a valence electron that was stolen. Okay. So our proteins, we need to make sure that we understand the important things about proteins and how they're different from other things. So they have nitrogen and they have sulfur. So if you're given a question in which you're supposed to pick out which one of these is a protein, okay, you're looking for the one that has the nitrogen and has the sulfur, because that's where we're going to see it. The sulfur will be in the R group. The nitrogen is going to be found in the amine group. That's part of your amino acid. Thinking about the structure of the amino acid, you've got a carboxyl group, so acetyl bonded OOH, and then there's a central carbon, then you have your amine group, and of course you've got your R group, and then a hydrogen, okay? And so the bond that's between our proteins is called a peptide bond. And I know I'm going through this kind of fast. I've got videos on just unit 1. So like you're able to watch those videos if you need to kind of get more information at any point. Okay. So there's going to be the peptide bond. There's a directionality to it. You've got an R terminus and a C terminus. R terminus just means the amine side of the long polypeptide. C terminus is going to be the carboxyl side of it, and it's going to grow from N to C. Okay. So in terms of our lipids, there's going to be 3 different types of lipids. You've got your steroids, you've got your phospholipids, and you have your fats. Your fats are going to have a glycerol with the 3 fatty acids. You've got your phospholipids that are going to have the phosphate group and then the 2 fatty acids. The way that they're going to associate themselves is that the phosphate group is going to be polar, is going to have the fatty acid tails, and so it's going to make up that membrane, okay? So it kind of makes the phospholipid bilayer, and so you're going to see the non-polar region of it is going to be the interior, and you're going to have the phosphate groups on the outside. Because of the fact that the fatty acids are so large, the entire membrane is considered non-polar, but it's going to actually be antipathic because there's that polar region and the non-polar region, okay? Because of the fact that the interior of the membrane is nonpolar, like our steroids are able to pass through. Small and nonpolar, so oxygen, carbohydrates, those things can pass directly through without any type of transport protein, which we'll get into in unit 2. And then our last one we have with the steroids. The steroids are just 4 fused rings. We're going to see them in signaling. So in cell communication, you might talk about a hormone, and that the steroid hormone, how is it going to, where's the receptor going to be? The receptor is going to have to be on the inside of the cell, because of the fact that the steroid is going to be able to pass directly through that membrane and get to that receptor on the inside. Okay, nucleic acids are our DNAs and our RNAs. The big things that are different between those, the DNAs are going to have deoxyribose, while RNA is going to have ribose. There's going to be missing an oxygen on the second carbon in the pentose sugar for the deoxyribose, while it has all of its oxygens in the ribose. You're going to traditionally see RNA is going to be a single strand, while DNA is going to be a double strand. And then we're going to see that there's difference in the nitrogenous bases. So the nitrogenous bases that we'll see in DNA is going to be thymine, while in RNA we're going to see uracil. So if you have a multiple choice question and they have a whole bunch of T's, but they're asking you about RNA, just go ahead and cancel those ones out. Okay. I mentioned about the monomer for the for the amino acids. For the proteins being amino acids. So in terms of nucleic acids, their monomer is a nucleotide. So you have a pentose sugar, that deoxyribose or that ribose. On the first carbon, you've got your nitrogenous base. On the third carbon, we always talk about like that hydroxyl group. It's not really a functional group, but that's okay. And then, well, it is a functional group, but then we also have our phosphate. So you're going to have the nitrogenous base, pentose sugar, and then the phosphate is going to make up your nucleotide. Carbohydrates are going to be your sugars, so like glucose and as we said, the pentose sugar that's in our DNA and our RNA, that's a sugar. And so the bond that we're going to see between those are going to be glycosidic linkages. So there was a question I saw recently about alpha versus beta. Alpha linkage means that they're going to be on the same side, versus beta flips back and forth in terms of like our glucose molecule. So starch we're able to break down because they're alpha linkages, but cellulose we're unable to break down because they're beta linkages. The monomer for our carbohydrate is a monosaccharide. And all of these are going to, like the proteins, all of them will go through something called dehydration whenever we're going to take these monomers and put them together to make the polymers. Now, this is foundational information, so we're going to keep building upon that as we keep working through them, okay? So let's take a quick break. We have a multiple choice question, so you can go ahead, and if you can work faster than me, you can go ahead and answer in the chat what you think the answer is. So scientists examined this folded structure of a purified protein that resuspended in water and found amino acids with a nonpolar R group were primarily buried in the inside of the protein. So what we see is that there's different layers to our protein folding, okay? And so the way that they're going to orient themselves is that we have our nonpolar our groups will be kind of facing toward the interior, and the polar groups will be facing the outside, okay? So the different levels to our protein, we've got our primary structure, which is all the amino acids together. Our secondary structure is our alpha helix and our beta pleated sheet, which is due to hydrogen bonds. And then our tertiary structure is going to be due to the R groups, where they're going to make covalent bonds, ionic bonds, hydrogen bonds, van der Waals, hydrophobic interaction, whatever we have. And then our quaternary is multiple polypeptides together. So this question is kind of asking us, why do we see the non-polars going inside while the polars going outside? This is really what we're seeing, okay? And so back to a question. Whereas amino acids with the polar R group were primarily on the surface of the protein, which is only best explains the location. So why are we going to see that the non-polar inside of the polar outside? Well, we have to think to ourselves, well, what does polar mean? Polar means that there's going to be polar covalent bonds. Because there's polar covalent bonds, we're going to see that there's going to be unequal sharing of that valence electron, okay? And so since there's unequal sharing of the electron, there's going to be a partial positive and a partial negative, which screams to me hydrogen bonds, okay? So let's look through our answer choice and see if there's anything about hydrogen bonds or about the ability to make those bonds, okay? So A, polar R groups on the surface of a protein can hide or form ionic bonds. That is not true. They're not going to make ionic bonds. Ionic bonds is a complete positive and a complete negative, and those are, again, attractive bonds. B, since polar R groups are too bulky to fit in the middle of the protein, so we're not looking at any type of bulkiness here, because we're looking at the polar versus nonpolar. These R groups are similar in size. Nonpolar R groups cannot form hydrogen bonds with water or push into the middle of the protein. This is why we see oil kind of floating on the top of water. Whenever you see oil and water mixing, they kind of like float at the top because they can't form those hydrogen bonds. And then D, of course, talks about nonpolar R groups that are different parts of the protein form covalent bonds with each other to maintain that protein structure. Okay. And I just realized I didn't say something earlier about nucleic acids. So I mentioned about the directionality that we have with our proteins. There is a directionality with our nucleic acids in which we have a 5' end and a 3' end. The 5' end is going to be where we have our phosphate group, and the 3' end is where we're going to see our hydroxyl group, the hydroxyl group that is on the sugar. Okay, and so DNA and RNA is going to be made 5' to 3', and we're going to keep getting through that. But I just want to make sure that I did mention that directionality that we have for it. Whew. So that was unit 1. Let's get into unit 2. And as I said, this is a cram session. I'm going as fast as I can to make sure that we can kind of get as much as we can in as much time as we can. And I know y'all don't want to hear me rabble along. Okay. So in terms of organelles, okay. So they're not going to ask you a straight question of what does the mitochondria do, and do not ever tell them that's the powerhouse of the cell. Oh my gosh. Anyway, so they're not going to ask you specifically what does it do. They're going to kind of ask you that, oh, there was this thing that was damaged. There's this problem here. And because of this problem, we're seeing that which organelle was affected because of this issue. Okay, so I've seen before with like Tay-Sachs that they talk about that there's a non-functional protein in this hydrolytic enzymes, which organelle is affected, and you're like, oh, well, that must be the lysosome because the lysosome is responsible for breaking down. Or there was a question on the release exam about the immune system and macrophage engulfing something, and which of these answer choices shows what happens when it engulfs, and you would have to be like, oh, the lysosome is responsible for digestion, so of course it went to that lysosome. Okay. And so quick rundown, our nucleus is where we're going to see our DNA. That's where we're going to see replication, replication and transcription are going to take place in the nucleus. And then, of course, that RNA is going to leave the nucleus to go out into the cytosol. We've got ribosomes in our cytosol. The ribosomes are going to be responsible for protein synthesis. And so that's where the translation is going to take place. We have our rough ER. Rough ER is where we're going to have those ribosomes once they get the signal peptide to move to the membrane so that we can make the proteins that are responsible for secretion as well as membrane proteins, so proteins that are sitting in that membrane. We do have our smooth ER. The smooth ER is responsible for detoxification. And so it's also for calcium storage. And so calcium is going to be a secondary messenger. And so we're going to see that we have that calcium that's going to be kind of responsible for that secondary messenger and releasing that information out. Okay. We do have our mitochondria. Mitochondria is where we see the site of oxidative phosphorylation as well as our Krebs cycle. So in the matrix is where we see the Krebs cycle. And then in like the cristae of the mitochondria is where we're going to see the oxidative phosphorylation. And yes, I know you're going to make a 100 on the exam. We also have our Golgi. Our Golgi is going to be responsible for packaging and modifying the materials that come from that rough ER. Okay, and then we have our peroxisomes, and we that are going to be for detoxification in forms of making the hydrogen peroxide. We have food vacuoles that are going to be made when you go through phagocytosis and bring something into your cell. The plant cells are going to have contractile vacuoles. I'm sorry, they're going to have central vacuoles to store the water and other waste products. And then we also have contractile vacuoles that are responsible for pumping out water. A lot of our organisms like protozoa that live in freshwater environments, water is rushing into their cells. And so they need to have the contractile vacuole to pump that water out so the cells don't burst. So we had this question on, I can't remember what year, but it's not that important, in which they're asking you to draw. So this is how we would maybe see question 5 on the exam, is that you would have to annotate a diagram. So drawing of this diagram, okay? And so what we're going to see is that where's the information? So if they want to know the path of gene expression, so the DNA is going to be in my nucleus. And so we go from DNA, we go through transcription, we then make the RNA, and the RNA is then going to leave the nucleus and it's going to go to a ribosome. In this case, since I'm making a membrane protein, it's going to leave and go to a ribosome that's found on the rough ER. Okay, so the rough ER is then going to go through translation. We synthesize that protein, and then that protein is going to go to the Golgi for packaging and modification. So that's where we're going to modify it slightly. We'll add some stuff. It then kind of figures out where does this protein need to go, and then it puts it in another vesicle, and the vesicle is then going to move to the plasma membrane. Okay, notice that I stopped the arrow at the plasma membrane because of the fact that we wanted to make sure that we went to the membrane because it's a membrane protein and we did not go past it. So be very careful when you're doing diagrams that you are very clear about where you're drawing the arrows to go to. Okay, so in terms of the other part of Unit 2, this is about kind of membrane transport. How do things get across the membrane? So first, let's think about what does the membrane look like. So as I said before, it's made up of phospholipids. So it has these little phospholipids. There's a phosphate group, and there's your fatty acid tails. The tails can either be saturated or unsaturated. If they're saturated, it means that there's no double bonds. If they're unsaturated, it means there's at least one double bond. It causes a kink in the tail, and so they don't compress as easily. Okay. We also have cholesterol in there. Cholesterol is in there. It's responsible for maintaining the fluidity of the membrane and making sure that we kind of can keep it fluid because we need to make sure that things can come across our membrane, as well as if it gets too rigid, it could break and it's problematic. Okay. We also have transport protein. So this would be considered a channel protein because it's like a protein that kind of goes across the membrane and materials can flow through it like it's a tunnel. Okay. We also could have carrier proteins. Carriers are going to bind, and then that causes a conformational shape change, which then allows it to move across the membrane. We also have glycolipids and glycoproteins. These are going to be responsible for like cell-to-cell communication and being able to recognize our cells. Okay. We also have, I'm going lots of places. Anyways, so in terms of our membrane transport, there's different types. You could have passive transport. Passive transport means that I'm not requiring any type of energy. It is going to move straight across that membrane. Okay. So there's two types. We've got simple diffusion. Simple diffusion goes across without the use of any type of resources. So like water can smidgen through, not a lot, but a little bit can smidgen through. But we're mostly looking at like our steroids. We're looking at like oxygen, carbon dioxide that are small and nonpolar. We want things that are nonpolar to get straight across. And yes, Fibrable is another resource. You could also use know it, like know and then a T at the end. They've got resources out there too for you. Okay, so that was our simple. We could also have facilitated diffusion. Facilitated diffusion means that there's going to be some type of channel protein or a carrier protein. And so facilitated means that it's still going down its concentration gradient. We're still going from high concentration to low concentration. It still doesn't require any type of energy. But what's happening is that it needs a protein to get across the membrane. So that's going to be usually our polar materials. Okay. And then active transport requires to have some type of energy. There's going to be ATP in some way, shape, or form. So that would be like our sodium potassium pump. That would be like a proton pump. Usually the word pump signifies that we're in active transport. And so that means it's going to go against its concentration gradient. So like, for example, if you're at the top of a hill on your bike, you could go right down that hill without any type of energy, right? But then if you had to get back up that hill, you're going to have to pump those bikes. So that's active transport. So if we go from low concentration to high concentration, that's active. We need to input some type of energy in order to get back there. Okay. And so that's usually going to be like materials against the concentration gradient. Okay. We also have active transport in terms of like bulk transport. So you have pinocytosis and phagocytosis. Phagocytosis is going to be cellular eating. So we're going to have these little pseudopods. They extend out, they surround the material, and then they make a food vacuole that then fuses with the lysosome. Pinocytosis, when you drink a lot, it makes you pee. No. So that's going to be like trying to gulp in extracellular fluid, which brings in a lot of our solutes. So here's our paramecia. It's a unicellular protist that has contractile vacuoles to remove excess intracellular water. In an experimental investigation, the paramecia replaces salt solutions of increasing osmolarity. The rate at which the contractile vacuole contracted to pump out excess water was determined and applied against the osmolarity of the solutions, as shown in the graph, which is almost the correct explanation for this data. Okay, so what we have here is we've got this line, right? And this is showing us that as osmolarity increases, the rate of contractions decreases, okay? So I'm noticing that correlation. I'm seeing that it's got this negative interaction. And so I'm gonna make myself a note. As osmolarity increases, rate of contraction decreases. And that's gonna kind of help me to think this through, okay? And so as I look at my answer choices, I'm kind of looking for that to be there because that's the answer that I come up with on my own, okay? So as higher osmolarity over here, lower rates of contraction are required. Oh, that's right. Because more salt diffuses into paramecia. But it's talking about where we're pumping out excess water. So we're not looking at that the salt is diffusing. We're looking at that there's an excess water. In addition, that salt is a charged ionic material. It's not going to pass across that membrane without some type of transport protein. So we're not going to see the salt just flowing into my cell like that. Okay. The contraction rate increases. Cool. As osmolarity decreases, cool, because the amount of water entering the paramecia by osmosis increases, okay? So the osmolarity is looking at the concentration of the solution. So as I move this way, I'm moving into a more hypotonic environment, which means there's more free water, and that free water can move across that membrane, okay, to where there is, of course, a higher concentration, okay, which that is my answer. Then we look at C, the contractile vacuole is less efficient at higher osmolarities because of a reduced amount of ATP. There's no information from this diagram that would make me think that. So I can't really think that it's going to be C, okay? And then D, an isoosmotic salt solution, there's no diffusion of water in or out of the paramecia, so the contraction rate is 0. At no point do I see that we're in an isoosmotic salt solution, so I have no way to know that. Okay, so that would, of course, be another answer that's not true, okay? And I'm seeing a whole bunch of B's popping up in the chat. Great job, all y'all. So we have another one, okay, and I didn't mention this earlier, and I should have mentioned, I'm sorry, we're just speed rolling, and that's kind of why the question's in there is to force me to talk about certain things. I kind of was strategic in this. So we've got these different cells, okay, and so they've given you the formula. Yes, you'll have the formula on the formula sheet. You're going to have to do a cube, a cylinder, You also could have to do a sphere and then a rectangular solid, if I remember correctly, or a rectangular prism, maybe. I can't remember. But I have math videos on all those. So if you're needing math videos on those, I do have those for you. Okay. Anyway, so I've got these different cubes, and they're asking you which of the following would be most efficient at removing waste. Okay, so removing waste means that I need to have a large surface area to volume ratio. So there needs to be a large surface area, and that large surface area is going to allow for the increase of materials to move across that membrane without having to move as far. Okay, and I love how people are going A and first one because I didn't really label them. So good job on y'all. And so like some students are like, wait, I have to now do all this math. No, y'all, you do not have to do the math. Okay, even if you want to do the math, cut that math. Make it 1, 2, 3, and 4 and see what the difference is. Do not try to do 10 squared. That's going to take you too much time. If you see math questions on it and you're able to kind of reduce the numbers, make them into smaller numbers to kind of help yourself out, do that. Do not use the numbers they have. That is a strategic way that you could try to do this math, okay? But if you did the math, you would have found this. You would have found 600, 1,000, and we're giving you a surface area to volume ratio of 0.6, okay? And so if you compare that to all the other ones, it's going to have the largest surface area to volume ratio with this first one. So that is going to be the correct answer on this one. Okay. You're all wrong. I'm sorry. They're all wrong. Okay. Anyways, sorry. I should have brought water up here and I did not. So Unit 3 is on our cellular energetics. Okay. So this is one of the larger sections that we see in our, on the exam. Remember, it was like the second highest percent or the third highest percent. And so this mostly is looking at enzymes, okay? And looking at inhibitors and competition and looking at the enzyme. How do we denature an enzyme, okay? So real quick, making sure that we understand this because my students mess this up and I want to make sure that you do not mess this up, is that if you get outside of the pH range, it's going to denature on both sides, okay? And so if it gets too low in the pH and there's too much H pluses, it will denature. If it's too high in pH, it's going to denature, okay? Now, the part that my students messed up, temperature, okay? If the temperature gets too low outside of the range, temperature gets too low, okay? It does not denature. The molecules start moving a little bit slower. And because the molecules move slower, the reaction rate is slower because of the fact that there's less kinetic energy and there's less of the collisions taking place, okay? Versus if we get into a high temperature condition, the particles are moving so fast, and because of the particle moving so fast, the bonds break and thus leads to a denaturation. So too low temperature is just decrease of rate because of the fact that the kinetic energy decreases, but if it's too high of temperature, it does denature them, okay? So when you think about those enzymes, you also think about competitive versus non-competitive inhibitors. Competitive inhibitors are going to bind to the same active site, versus non-competitive inhibitors are going to bind to a different site, both of which are going to change the shape of the protein, okay? So if you have anything bind to a protein, it will always change shape, okay? So that change of shape is what's going to lead to what we see happening afterward, okay? And that's why the enzymes work the way they do. My husband's trying to bring me water. It's kind of cute. Anyways, so I do not need water. It's fine. Thank you. So we have cellular respiration. This is going to take place in our mitochondria. Okay. And so, well, not all of mitochondria. So the first step of cellular respiration is glycolysis. Okay, so glycolysis is going to be where we have the sugar splitting steps. So we have our glucose, and we break the glucose into 2 pyruvates. Okay, we make 2 ATP, and we have 2 NADHs that are formed. This takes place in the cytosol, which means that all things can do this. Prokaryotes and eukaryotes can do this. In a prokaryote, they're usually going to go through fermentation afterward because of the fact that we need to regenerate that NADH. Otherwise, we run out of NAD pluses. In case you don't know what I mean by NADH, NADH is a material that is holding the electron to take it to the electron transport chain. And if I have all of them holding the electrons and there's nothing to take more electrons, I'm not going to be able to go through glycolysis. So the fermentation process is going to allow you to kind of let go of that electron. Is going to allow you to oxidize the NADH so that you'll be able to continue kind of breaking down that glucose, okay? So after we make the pyruvate, we go through pyruvate oxidation, which, did I give my, yeah, I did. Okay, so after we go into pyruvate oxidation, this is where we're going to go from pyruvate into acetyl-CoA, and then it's going to enter into the Krebs cycle. The Krebs cycle is where we're going to finish the breakdown of our, the glucose, okay? There was a question in the chat, the difference between osmolarity and osmolality is just about the units. Osmolarity is using kind of moles into liters, and osmolality is into kilograms of material. Somebody asked me that recently. Anyway, so back to the Krebs cycle. So we're gonna make ATP, we release the carbon dioxide. So at this point, we've released all the carbon dioxide, which means that all of our glucose has been broken down. We make NADH and FADH2, okay? You're not going to be asked the specific numbers of any of these, okay? If we ever see a question, it's usually them giving you a diagram. On the mock exam, there was a question where they gave you the diagram of glycolysis and asked you how much ATP is made. Do we know that answer? Yeah, we know that's 2 ATP, but they also gave you a diagram. Okay, I'm sorry your brain hurts. I'm going as fast as I can to make sure that you can get out of this soon. And then the NADH and FADH2 are then going to go to the electron transport chain. The whole function of the electron transport chain is going to be to allow these high energy electrons to release that energy, which pumps a proton, okay? So it's going to pump that proton into the intermembrane state. So you see how we have these two different membranes? The mitochondria is double membrane, okay? So it has the inner membrane, and it has, of course, its outer membrane. So we're going to pump those protons into this intermembrane space, so it leads to this high concentration, so it's a low pH, okay? And that is electron transport chain. Oxidative phosphorylation then also has the chemiosmosis, which is where we allow ATP synthase. It's going to be a protein, and as the proton binds to it, it changes shape, which allows it to kind of move like a rotor almost, and it moves the proton back across the membrane, which allows for the phosphorylation of ADP to make ATP, okay? Now, again, the citric acid cycle, Krebs cycle, and the oxidative phosphorylation is taking place in the mitochondria. So if you don't have a mitochondria, you traditionally don't go through these steps, okay? Photosynthesis has two steps. You've got the light reactions. Light reactions are going to be here. You've got the light shining down on your photosystem. Photosystem 2 is first. That causes this electron to kind of move to a higher energy level, and it goes down to an electron transport chain, again, pumping protons across that membrane, okay? So when the protons move across the membrane, they move into the thylakoid space, and then later on, we'll go through ATP synthase so that they can synthesize ATP. So we'll synthesize ATP between photosystem 2 and photosystem 1. The electron then goes into photosystem 1, gets more light, which then allows it to go to NADPH. NADPH, again, is holding the electron. The way you remember this is that the P is for photosynthesis. So NADPH is going to be found in photosynthesis, while NADH is not. So they can sometimes trip you up on a question by, if it's about photosynthesis, they could put NADH as an answer choice, and you should know that that is not the right answer because that is in cellular respiration, okay? Versus NADPH is in photosynthesis, okay? And so we also have the water splitting step. It's called photolysis. This is where we're going to break down that water to regenerate that electron, which I didn't mention before, but at the end of the electron transport chain, there's an oxygen here that takes that electron and it forms water. And so it's just the reverse of what we saw in the electron transport chain of cellular respiration taking place in photosynthesis, okay? And that's what gives the electrons to this process, okay? So the ATP and the NADPH then goes over here into our Calvin cycle. The Calvin cycle is going to start with our carbon dioxide. We use the enzyme Rubisco and RUBP, and then we're going to synthesize something called G3P. If you really looked at the steps of glycolysis, you would see that there's actually 2 G3Ps. It's an intermediate step before it moves into pyruvate. So it's actually, I think, like the 5th or 6th step of glycolysis. But so this is just to show you that we have the same intermediates in these two processes, but they're not exactly reverses of one another. And then you will regenerate the RUBP because the cycle you have to restart everything again, okay? And so notice here that there's 9 ATPs that are used, but there's only 6 NADPHs that are used. The reason why is because we have 2 different ways the electron can flow through the light reactions. So we have linear electron flows, which I explained, okay? So we go from PS2, we go through the ETC, PS1, and then we have the NADPH. So it forms a 1-to-1 ratio between ATP and NADPH. Cyclic electron flow is going to go from here from photosystem 1, and it comes back down this electron transport chain. So they can generate additional ATPs without generating the NADPHs, okay? That was on the AP exam last year. I think it was question maybe 4, in which they asked you about that process. They gave you the diagram, and then you had to apply it, okay? Now, the diagram looks slightly different, okay? So just be careful that when you see a diagram on the test, you don't freak out. They're doing their own drawings, and because they did their own drawings, it's going to look slightly different, okay? So where are we at? So also don't get stuck on like all the minor details. So kind of just keep in mind what's going in, what's coming out, where it's taking place, and why it's important, okay? So this was the 2015 exam, okay? Now, I know it's on a different CED, but still, yes, this is available after live stream is over. So here's our cellular respiration. It's going to show you all the different steps, and so you just had to apply it, okay? The question was asking a contribution of each and how that makes ATP. So if I look here, I can literally see ATP in this diagram. I know that ATP is formed, okay? So you could talk about the ATP is formed by attaching, like by substrate level phosphorylation. You could also talk about that we make the NADHs. And then if you look at the diagram, you can see that acetyl-CoA is also right here and is in the previous diagram. So you can talk about how the acetyl-CoA is moving into the Krebs cycle, okay? You could use the diagram to answer the question. So use those diagrams whenever you can use those diagrams, okay? So the second part was asking about the intermediates of the Krebs cycle. Again, we're making the NADHs that's going to go to the electron transport chain, the FADH2 that will go to the electron transport chain. And I think they're even shown in the picture. See, you can see the NADH there. So you could say that the NADH was formed and bringing these high energy electrons to the electron transport chain. So you can literally see it taking place in the picture, which helps you to explain it. You also see that there's GTP, and GTP is formed by substrate level phosphorylation. And in case you don't know, ATP is adenosine triphosphate. GTP is guanosine triphosphate. Instead of it being an adenine on this nucleoside, it's a guanine. It's basically the exact same thing. We have TTPs and CTPs also, okay? So you could have mentioned any of these, about the high energy electron for electron transport chain, NADH and FADH2. Talk about pumping the protons against concentration gradient when you get to electron transport chain, or talking about the GTP is for the substrate level phosphorylation. Okay. So in terms of our forming of our proton gradient, okay, so we can form this gradient. The reason why is because we have these high energy electrons, and it's going to allow us to form that gradient. Okay. So what does that gradient do? This one requires you to know something. Okay. We're going to use that gradient. It's going to go through ATP synthase to synthesize ATP by oxidative phosphorylation. So it provides the energy for oxidative phosphorylation of ADP, and then we're going to have the flowing of those protons through that ATP synthase to generate the ATP. Okay, so they then go on and they want you to justify. So that's the thing you're going to see a lot in these exams. Okay, is justify something. Why do we see this? Well, all organisms perform glycolysis. Well, why do all organisms go through glycolysis? Well, all organisms go through glycolysis because it must have taken place when there was no oxygen. Sorry, I'm ahead of myself. And when there was no mitochondria. So it precedes this mitochondria, okay? Also talking about that we had evolution in which it was a selective advantage. Those that could go through glycolysis were able to survive, okay? Then you talk about anaerobic, the fact that it predates the atmospheric oxygen. It doesn't need oxygen for this process. And then it only occurs in the cytosol because it predates our membrane-bound organelles, okay? So, yes. And then they had you do math. Yes, you're going to have to do math, and it may not be math that's on your formula sheet. Yes, that's the reason I put this question here, is because this is math that isn't on a formula sheet that you would have to come up with. So they tell you the researcher estimates in certain organisms that there's complete metabolism of glucose produces 30 molecules of ATP. So when I break down glucose, I get 30 ATPs. Cool. The energy released from the total oxidation of glucose under standard conditions is 686. Okay, so if I was to figure out how much energy is in glucose and I oxidize it completely, there's 686 kilocalories per mole in there. Okay, but ATP to ADP is only 7.3 kilocalories. Okay, so I have to calculate the amount of energy that's available from the hydrolysis of 30 moles of ATP. So how would I do that? Well, I know that there's 30 molecules of ATP. Each of them is 7.3, so I'm just going to multiply. And that gives me 219 kilocalories, okay? And the reason why it's a kilocalorie is because the mole and the mole cancels out, because you have a mole in the numerator and a mole in the denominator, and they cancel, leaving you just with the kilocalories, okay? So then it asks you to calculate the efficiency of the total ATP of 1, okay? So the efficiency means how good is it, okay? Well, we made 219. We should have had 686. So the efficiency is going to be 219 divided by 686, which means it's 0.319. So it's either 31% or 32%. And yes, you do have a formula sheet on the exam. It's the first page of your booklet. That formula sheet is actually going to, you can't rip it out, but it doesn't have all the formulas you might need because they assume that you can do dimensional analysis, which is what this is, okay? So you got 31% or 32%, and it said describe what happens. It's going to be lost in the form of heat, okay? I'm sorry that's chem. Dimensional analysis is not just chem. It's how you can convert units. And yes, you'll be able to use a calculator. It's a TI calculator, any of the calculators you want. There are certain ones that aren't accepted, so just check College Board website about which calculators you can use, okay? So now I know this is calculate. I always tell my students write this in complete sentences. I would just say the efficiency is 32%. The amount of energy is 219. It does not take you that much longer to write 3 more words and make it into a sentence, okay? So on to unit 4, and it is almost 53 minutes. I'm so sorry, y'all. So cellular communication, okay? If people keep asking me, what do I predict is going to be on the exam? I predict there'll be a cell communication question. There's going to be some type of diagram that you're going to have to analyze. And yes, TI-84 will be allowed on the exam, okay? And so the 3 steps of signal transduction is you have your receptor, transduction, and then your response. The receptor is shown in the picture. You can see it's a G protein receptor. You can see that the ligand binds to the receptor. Because of the fact that this is a membrane-bound receptor, that tells me that my ligand must be polar because it can't get through that membrane. If it was non-polar, it would be found inside. So that's a question they sometimes might ask, okay? So once the ligand binds to my receptor, it's then going to cause something else to happen. So I can see here's step A and then step B, it's bound, and that activates this adenylyl cyclase, okay? I then see the adenylyl cyclase is going to go make the ATP into cyclic AMP, which then activates my protein kinase, which then causes the chlorine to be secreted from the cell, okay? So they're showing you this pathway, and then they're going to ask you, what happens if? So what happens if the ligand can't bind? What happens if there's a mutation that keeps something on? What happens if, okay? So what happens if the adenylyl cyclase could not become activated? What happens if this step does not happen? Well, if this step doesn't happen, then that means we're not going to see cyclic AMP being activated. Okay, we won't see the ATP making into cyclic AMP. Well, if we don't see the cyclic AMP becoming formed, that means that we're not going to see protein kinase becoming activated. And if protein kinase doesn't get activated, I don't secrete chlorine. If I don't secrete the chlorine, then I'm not going to see water being released out of my cell. Okay, the question was actually about cholera and about the fact that you lose your water in cholera if you have a cholera infection. And so they were asking, well, why? And so this was the reasoning why. It's because the chlorine gets pushed out, and because the chlorine gets pushed out, it makes it hyperosmotic or hypertonic outside, and the water follows it out, which is why you have very runny stools when you have cholera, and you become very dehydrated, okay? So other things that we have in Unit 4 is on our checkpoints, okay? So when we think about the cell cycle, okay, let's first talk about cell cycle. Actually, I think I'm ahead of myself. Yeah, let's talk about the cell cycle, okay? So in the cell cycle, we've got the G1, which is where the cell's going to grow. We have the S, which is our DNA synthesis. This is when we're going to go through replication and make a copy of that DNA. Then we go through cell growth. Again, the G2, again, cell grows, okay? So we have a couple different checkpoints, okay? The first checkpoint you're going to have is the G1 checkpoint. This is making sure, should I divide? Should my cell go through division? Should I make a copy of this cell, okay? The DNA synthesis then is where we're going to synthesize that DNA. And the G2 is going to make sure, A, did I completely make a copy of that DNA? And the copy that I did make, okay, is there any major errors to it? So it's going to check to make sure that everything's good, okay? And then we go through mitosis, okay? Mitosis is just going to be nuclear division, so dividing of that nucleus in half, okay? So we have the M checkpoint, which is really cool because it takes place in the metaphase, which is when the chromatids are lining up on that metaphase plate. So since our sister chromatids line up on that metaphase plate, this is going to ensure that we have every single microtubule attached to every single sister chromatid to ensure that when I do go through anaphase and I separate them, that I'm going to get one set of chromosomes to go to one side and one set of chromosomes to go to the other. So the M checkpoint ensures that we get equal division of our nucleus, okay? So I did mention a couple words just a second ago, and I'm so sorry. So what we did with the G1, the S, and the G2, that was a little interphase. That's kind of in between cell division. And then mitosis is when we're going to see the actual nuclear division, so the division of the nucleus, okay? So we go through the interphase. This is when, cells growing, we're making a copy of our DNA. Then we go into prophase. Prophase, you see the nuclear envelope is kind of disintegrating. We're getting rid of the nuclear envelope, okay? You also see that your spindle fibers are moving to the opposite sides to kind of get prepared. And in metaphase, that's when we're going to see that tug of war. So we have these microtubules. Microtubules are going to be attached to kinetochores that are going to be on the centromeres of the chromatids. And so that's going to attach, which then kind of they tug of war. And as that tug of war, it lines them up in the middle, which is our metaphase. You can remember like M for metaphase. And then anaphase, when we see the sister chromatids are going to be pulled apart to opposite poles. And then we have telophase, which is where we're going to see our two new nuclei. Okay, some people may call that telophase, but I do not call it that. I call it a telophase, but that's okay because it's a telephone, but whatever. And that's when we make our two new nuclei. And then we go through cytokinesis, which is when we're going to divide the cytoplasm. Okay, so from prophase through telophase, this is all mitosis. Okay. The cytokinesis will take place after mitosis, and the interphase takes place before mitosis. Okay, and so a couple years ago, I was in my student teaching, or maybe more than a couple, but whatever. And they did this thing called the cell cycle dance, okay? And so we've got, of course, we're going to think just on mitosis, okay? So we have prophase, metaphase. Actually, let's just use cell cycle. So G1, S, G2, prophase, metaphase, anaphase, telophase, cytokinesis. Yes, I really just did that. I just want to make sure the students got it, Penguin. That's okay. But anyway, so we go through that where we had the G1, where we had this is the DNA. And then we go through S phase where we're making a copy of that DNA. And then G2 is just when it grows. Prophase is when we were preparing to divide. The sister chromatids were going to condense. And because those sister chromatids condense, they kind of find each of the sister chromatids together and they pair up. And then we went into metaphase. Metaphase is where we're going to see that we've got the sister chromatids lined up in the metaphase plate. Then the anaphase is when the sister chromatids were moved apart. Telophase is when we had our two new nuclei. And then cytokinesis when we divide the cytoplasm. Okay, that just woke you up. Anyway, so what they might do is they might ask you questions about, like, I don't know, there's cancer drug out there, and the cancer drug inhibits the production of the microtubules. And so they're going to say, well, when would it be arrested? When is it going to stop? Okay. And so it's going to stop like right here because of the fact that we're not going to be able to go through anaphase because of the fact that we don't have those microtubules formed. We're inhibiting them from taking place. Jump scare warning. Anyway, so insulin is a protein hormone. This is secreted in response to elevated blood glucose levels. When insulin binds to its receptors in the liver cell, the activated receptors stimulate a phosphorylation cascade that causes translocation of glucose transport to the plasma membrane. Based on the information, which of the following best describes the role of insulin in this liver cell signaling pathway? Okay, so let's look at this. It says we have a protein hormone. Protein hormone means that it's going to be a polar material. So the membrane receptor, okay, it then says it binds the receptor on the cell. Okay, cool, I knew that. It then says that we stimulate a phosphorylation cascade. Phosphorylation cascade, then that part of transduction, and then that causes translocation of glucose. Well, that's my response. So my signal is the insulin, then the phosphorylation cascade is my transduction, and then my response is translocation of glucose, okay? And so it's asking me what is the role of insulin? Well, it's going to be the signaling molecule. Which one is a signaling molecule? Is that a ligand? I don't know. Is it a receptor? No, it says it binds the receptor, so that's not it. Secondary messenger. Secondary messenger would have to be something that happens second, so that can't be it. And a protein kinase, I know that's responsible for a phosphorylation cascade. So the only logical answer we have is, of course, our ligand. Okay. So great job, everybody who put that on there. This one is actually in the, I'm going to have myself. This one's actually in the CED. So it could be like that. And this is what we call one of those independent questions. It's just a little bit of information and then you're answering that question. Okay. And so moving into unit 5, and I'm so sorry that it's 4 o'clock, but I'm moving as fast as I can. I just want to make sure I give you as much of a review as I can. If you have to leave because it's 4, I completely understand. This recording will be available on my website. It'll be available on my YouTube. It will be available for you that you can come back and catch it later if you needed to catch it later. Okay. And so in unit 5, this is where we get into meiosis. Okay, so meiosis is going to be when we're making our gametes. Okay, so our sex cells. Okay, and so we're going to go through 2 rounds of division, but only 1 round of replication. So here you can see that replication that's taking place in our interphase. We make a copy of it, and now we're going to talk about something called homologous chromosomes. So before we were looking at sister chromosomes. So in mitosis, we're all looking at the sisters. Now that we're in meiosis, we're talking about the homologous chromosomes. Homologous chromosomes means that I have a maternal set and a paternal set. So I've got my mom's genes and my dad's genes. Okay, or the copy of my mom's allele, my dad's allele. Okay, so what we'll do is we go through 2 divisions. The first division is our meiosis 1. We go through prophase, metaphase, anaphase, telophase, and again cytokinesis to divide it. It's not actually called cytokinesis, but it's not really important to know what the name of it is, so don't worry. Okay, and then we go through the second round of division again: prophase, metaphase, anaphase, and telophase, cytokinesis. And so the way that we differentiate the first round over the second round, we just put a 1 behind it. So everything that will be in the first round division is going to be prophase 1, metaphase 1, anaphase 1, and then telophase 1, and then the second division is prophase 2, anaphase 2, yada, yada. You get the idea. Okay. So there's things we have to make sure that we understand about this division. Okay. So since we're now looking at homologous chromosomes, the homologous chromosomes are what's lining up on the metaphase plate now. So before we looked at mitosis, it was sister chromosomes aligned on the metaphase plate. Now that we're in meiosis, we're looking at homologous chromosomes are lined up on that metaphase plate. Okay. And so when we go through anaphase 1, we now make haploid cells. So the end product of meiosis 1 are haploid cells that have sister chromatids. So since I already have my sister chromatid, I don't have to go through division. I don't have to go through replication and make a copy of that sister chromatid. I will then go through the second round. And so the second round of division, meiosis 2, is very similar to mitosis, is exactly what we saw with the sister chromatids, okay? Now, there's some things that happen during meiosis that allows for the genetic variability that we have, okay? So the first thing is crossing over. Crossing over is going to take place during prophase 1. So when our homologous chromosomes line up and kind of like, not line up, but when they pair up in prophase, when we're condensing and kind of getting all together, you're going to see that your maternal chromosome and your paternal chromosome, they can overlap, okay? So this will be chromosome 1 from mom and chromosome 1 from dad. They overlap, and when they overlap, they can exchange genetic information. So this chromosome can be mostly mom with a little bit of dad, and this chromosome can be mostly dad with a little bit of mom, okay? And so you're able to get kind of slightly different information, okay? We also see independent assortment taking place in metaphase 1, okay? So in metaphase 1, we're going to see that we're going to have the homologous chromosomes line up on that metaphase plate. And so since the chromosomes line up on that metaphase plate, we're going to see that it could either line up where maternal's on one side and paternal's on the other, or it could be the opposite way where we have paternal and maternal, okay? So they can line up on opposite sides, and it's independent for the 23 different sets of chromosomes that we're looking at. So you get a lot of variety in your gametes, okay? And so there's that. And I kind of did the comparison as I was talking through that. So kind of big comparisons is that they both have one round of replication. Mitosis has one division, meiosis has 2 divisions. There's going to be crossing over takes place in meiosis that does not take place in mitosis. There's going to be independent assortment that takes place in meiosis that does not take place in mitosis. We're going to see division of sister chromatids in mitosis, and we're going to see homologous chromosomes in meiosis. We're going to see that the parents and the daughters in mitosis are genetically identical. We make 2 cells versus in meiosis, they are genetically distinct from the parent. The parent is diploid and the offspring are haploid, or the gametes are haploid, okay? So Mendelian genetics, okay, so this is going to be where we look at like complete dominance and incomplete dominance and all that stuff, okay? So first thing before we get too far into this, I want to make sure you understand that dominance doesn't mean prevalent. Okay, so dominance means that it's going to make a protein while the recessive usually doesn't make a protein. And so here we see this individual. Okay, count his fingers with me. 1, 2, 3, 4, 5, 6. I have 5. How many fingers do you have? Do you have 5? Anyways, so this is called polydactyly. This is a dominant trait. Okay, and so most people that I know have 5 fingers on each hand, okay? And so this is actually a genetic inherited trait, and it's really cool, isn't it, to have 6 fingers? Like, yeah, 6 digits. So just because it's dominant doesn't mean it's prevalent. You don't really see most people having that trait because most people are going to be homozygous recessive, and they're going to have 5 fingers on each hand and each foot, okay? 5 toes on each foot, yeah, whatever, okay? So we then want to kind of think about complete dominance versus other types. Okay, so complete dominance means that if you have one dominant allele, you're going to have the dominant trait. If you have 2 recessives, then you'll have the recessive trait. Okay, so that's really complete. So you can be homozygous dominant or you can be heterozygous and show the dominant trait. Okay, versus in like incomplete dominance. Okay, we have 2 dominant traits, and both of them are kind of incompletely dominant, okay? So they both make something. So this one makes a red protein, this one makes a white protein, and when red and white come together, they make pink, okay? And so this is going to be incomplete dominance, okay? And if you were to cross these 2 pink ones again, since they're heterozygous, you would get a 1 to 2 to 1 ratio, versus in our monohybrid cross, we get a 3 to 1 ratio, okay? So that's kind of showing you the differences in the ratio, okay? This next diagram is a comic. It's not true, but it does show what I want it to show, okay? So here I have a black bear and a white bear, okay? And when I cross them together, okay, I'm going to get a bear that has both white parts and black parts on it, okay? And yes, that'd be like a black bear and a polar bear, but those can't mate, okay? Anyways, and no, they don't make panda bears, okay? Anyways, so this is showing co-dominance, okay? So both of them are dominant, and you can see them individually as distinct pieces, okay? The same thing happens with your blood type, okay? So we have A blood type and B blood type, and you have AB. The persons that have AB blood type mean that they have both the A protein and the B protein on their red blood cells because they have this codominance, okay? Then we also kind of with this, they're usually going to be some type of pedigree. I can almost guarantee you will have a pedigree somewhere on your test, okay? So you need to be able to figure out whether it's dominant or recessive, okay? I have noticed lately that they've been like using male as an XY individual and female as XX. So don't just see the XX and the XY and assume that we're looking at a sex-linked trait because they have been kind of writing it differently just to be more inclusive, okay? And so what we're looking at is whether it's found in every generation. Okay, so if we look here, I've got 3 and 4 of the parents, and they have a child that does not, I'm sorry, that does have the trait, but they didn't have it. That immediately screams to me some type of recessive trait. Okay, and then when I look here, I can see that the father didn't have it, okay? The mother didn't have it, okay? I can see here that I have affected father, okay? I see that here I have a mother that's affected and a father that's not affected. 19 would not be affected if we were looking at sex linked, okay? Because of the fact that the father doesn't have a faulty X to pass it on. So we would not see sex linked recessive here because of the fact that he doesn't have that X to pass it on. And so we know that this is autosomal recessive, okay? So we also kind of think about non-Mendelian genetics. So non-Mendelian genetics is looking at like things that are extranuclear, so things that are not actually part of the nucleus. Okay, and so your mitochondria and your chloroplasts are believed to be prokaryotes, and so because of that, they have their own DNA. So with what's happening tomorrow, do you know what's happening tomorrow? I know what's happening tomorrow. Tomorrow is Mother's Day. So I figured I'd go ahead and say, hey, happy Mother's Day to all the moms in your life. Just remembered how much to thank them tomorrow because they gave you, of course, the X chromosome. They gave you the antibodies. They gave you oxygen. They gave you nutrients. But more specifically, in terms of what we're looking at, they gave you mitochondrial DNA. And so if you have a question, like last year, I think there was a question on mitochondrial DNA and about like these different plants and what color they were going to be. And the trait was a mitochondrial trait. You have to follow where the mom is. The mom is the one that has the mitochondria, and the mom is the one that's going to pass that on to her young. Okay. Every single young will get that trait from that mother. Okay. So make sure that you do understand those non-Mendelian things. We're looking at like mitochondrial and chloroplastal inheritance. Okay. So here we have a diagram. The following figure displays data collected from studying a family, some members of which have sickle cell disease, a rare genetic disorder caused by mutation in the hemoglobin beta gene, so HBV. There are at least 2 alleles of it, HBA and HBS. HBS causes sickle cell, while HBA is a wild type. Genetic testing then gives us insight. We have this beautiful little figure that we should probably look at a little bit bigger. So based on the figure, data in the show, which one best describes the genotypes of the individual family members? Okay, so let me look in here. Let me see. If I see here, I got these 2 parents are unaffected, but they have an affected child, okay? So that tells me that some type of recessive, okay? So that will tell me that my affected individuals are going to be HBS because of the fact that this is a recessive trait, okay? So they have to have 2 of these recessive alleles in order to have the sickle cell trait, or the sickle cell disease, okay? Versus those that are not affected are either going to be heterozygous and they're going to be considered carriers, or they'll have sickle cell trait instead of sickle cell disease, or they'll just be homozygous for the HBA allele, okay? And so we want to see which of these follows the genotype. All affected individuals possess at least 1 dominant allele. Well, if they're all affected individuals have 1 dominant. That's not right because of the fact that this one is homozygous recessive, okay? Healthy individuals may possess 1 mutant allele. Well, can I be healthy and still have the mutant? Well, yes, I can. Here I can be HBS, HBA, which means that I can have one of those alleles. Then it says that, 4 and 5, wherever you are, 4 and 5 must be heterozygous. They are both affected, which tells me that they are not heterozygous. And then 2 and 6 have 2 copies. They cannot have 2 copies because of the fact that this one was able to pass on that recessive allele. So I know that D is also wrong. So the answer had to be B. Okay. So unit 6, okay, this was where a lot of students have trouble. I know that for my students, this is one of their weakest units. Think about the difference between DNA and RNA. We mentioned this earlier. So DNA is going to have deoxyribose. RNA is going to have ribose. DNA is going to be double-stranded. RNA is going to be single-stranded. We're going to see that there is a nitrogenous base of thymine in DNA, and it's going to be uracil in RNA. And then we also can think about that they have different sugars. I don't know if I said this already, but they have deoxyribose versus ribose. Okay. And so the central dogma is made up of like how we see this gene expression. Okay. So we have our replication. So we make a copy of the DNA reading DNA. Okay. So you're going to read DNA. You'll always read DNA 3' to 5'. Okay, so we're going to start where the hydroxyl group is and go to where the phosphate group is. Okay, because DNA is anti-parallel. Okay, wherever you have a 5' group, you have a 3' group. Wherever you have a 5' group, you have a 3' group. They are anti-parallel. They're equidistant from each other. They're equidistant because they have a purine and a pyrimidine. In case I didn't mention this before, a purine means it's going to have a double ring nitrogenous structure, and that would be your adenine and your guanines. And then your pyrimidines are going to be a single ring structure, a single nitrogen ring. And that would be your cytosine, your uracil, and your thymine. Okay. So I always remember it as pure silver. So purine is Ag. And then you want to cut the pyramid, CUT, cytosine, uracil, and thymine. Okay. And so those are your big differences there. And then we're thinking about replication. We're going to read it 3' and we're going to make it 5' 5. We're going to read it 3 to 5, and we're going to make it 5 to 3. Okay. This will take place in our nucleus. We then go through transcription. Transcription is going to read that DNA, and it's going to synthesize an RNA strand from that. Okay. So before I forgot to mention it, DNA polymerase is going to be used to make that DNA polymer. We have helicase, which is going to break those hydrogen bonds that are between our nitrogenous bases. We also have topoisomerase, which is going to relieve the strand upstream. We have ligase, it's going to seal the linkage between them. So like when you go through it, replication, we've got a leading strand and a lagging strand. The leading strand is going to move continuously into replication fork, while the lagging strand is going to move discontinuously, so it moves against like the fork. So we open up a little bit and it makes a little strand, and it opens up a little bit more, makes another strand. It keeps going against the replication fork. And so we have to put those Okazaki fragments together using ligase. So that was replication. So transcription, we read the DNA, again, 3 to 5. We synthesize the RNA 5 to 3 using the enzyme RNA polymerase. It's going to pry apart the two strands, makes a copy, and as that copy, it kind of falls out to the outside. And so when we go through translation, we're going to read that mRNA, and we're going to start at the start codon being AUG. You do not have to know the codon chart off the top of your head. You do not have to have it memorized. It will be given to you on the test, I promise. And so then you will look at each of the groups of 3 your codons to see what that correlates to in terms of your amino acids. So AUG is methionine, AUC is isoleucine, and UCG is our serine. And then, of course, we have UAA, which is our stop codon. You do not have to know the stop codon, but of course, if you are a fan of UGA, you know they're unstoppable. And so you can remember that UGA is one of your stop codons. There's some things that happen after you go through transcription in a eukaryote that does not happen in a prokaryote. In terms of eukaryote, we go through splicing, and we cut out the different introns. So these different non-coding regions can get cut out. And then we can add a 5' cap. So there's a guanine cap added to the one side. And then there's a poly A tail, so a tail full of adenines. The guanine cap allows it to facilitate movement outside the nucleus, as well as it allows for the ribosome to know where to bind for translation. And the poly A tail is going to protect it from the hydrolytic enzymes that are in the cytosol. They're going to try to shorten your mRNA strand. Okay, there's different mutations that can take place. A silent mutation is going to be if we're talking about a point mutation, it's going to be one nucleotide substituted for another nucleotide, but it still makes the exact same amino acid, so it's silent. You don't even notice it. Okay, versus a missense mutation. It's like at the beginning of the thing, which I don't even know if you noticed, but I mentioned that you could go walk your dog, or I said you could walk your fish. That doesn't really make sense in the sentence. Now, fish is actually an animal. That's logical in that sense. But why would you go walk your fish? Okay, so that's a missense mutation. You have one amino acid substituted for a different amino acid. And then nonsense would be if you just have a random stop codon in the middle of your sentence. So if I was in the middle of talking, all of a sudden I just stopped talking, that would show the nonsense. So my protein just prematurely stopped. Okay. We also have frame shift mutations. If you have a deletion of 1 or 2, or you have an insertion of 1 or 2 nucleotides, it shifts the group of things. So instead of you saying the cat ran too far, you read to cat ufa, which doesn't make any sense. So it's making sure that you have the right reading frame. And so if you add or subtract 1 or 2 nucleotides and you mess up that grouping of 3, you could mess up the reading frame, which then leads to something different. Okay. We also have biotechnology. Biotechnology is where we're going to see where you have the gel electrophoresis, and you've got your PCR, and you've got your transformation experiments, all of those type of things. So with the gel electrophoresis, what's going to happen is we're going to cut the DNA with restriction enzymes, and then we're going to put it into a well. And so then we'll turn electricity on, and the smaller fragments are going to get pulled toward that positive region faster than our longer fragments are. So they're going to be pulled toward the positive end because DNA is negatively charged. In case you don't know that, DNA is negatively charged because it has a bunch of phosphate groups. Phosphate is negatively charged. It's the PO42 minus. And so because of that negative charge, DNA and RNA are, of course, negatively charged. And so they're being drawn toward that positive end of the gel electrophoresis. Okay. If you see a thicker band, that means that there's more DNA there. If there's a thinner band, that means that there's less DNA there. And so oftentimes they could tell the difference between something that's homozygous or heterozygous, because in fact you'll have more bands with the heterozygous individual because they were cut differently. And then the homozygous is going to have the same band of the same DNA, and it's kind of buckled on top of each other, making it a thicker band. Okay. PCR is polymerase chain reaction. We're going to go through and add the DNA, and it then makes a chain reaction and makes multiple copies of it. And then transformation experiment is when you transform bacteria. So y'all add the plasmid to bacteria, and then you made it antibiotic resistant. So they asked you questions based on that. Okay. So here we have this genetic disorder. It alters our glucose metabolism of first symptoms in adulthood. We have this family that shows different generations 2 and 3 are known to have the disorder. Based on the pedigree, the geneticists concluded that the disorder arose in individual 2 and 3 was caused by mutation in mitochondrial DNA. That's important for us to know. Okay. They then gave us this data, and then they were telling us when individuals are normal, at risk, or affected. Okay. So part A on these FRQs, this is, I think, example number 2. So this is going to be a graphing question. It's going to be a biology question. Do you know biology? Okay, so the disorder alters glucose metabolism. Describe the atoms and types of bonds in a glucose molecule. So you have to think to yourself, okay, what's glucose? Glucose is a carbohydrate. The different things that are in a carbohydrate. I've got the I've got the carbon, and I've got the hydrate. So hydrogen and oxygen. So the 3 different atoms that make up a carbohydrate is a carbon, hydrogen, and an oxygen. Whew, got that. Okay. I then have to come up with a bond. Okay, what kind of bond? Glycosidic linkage is a bond. What's the glycosidic linkage? Glycosidic linkages is between glucose molecules. So that's not right. I want in a glucose molecule. That would be covalent bonds. So there's a covalent bond between the carbon and the hydrogen, and there's a covalent bond between the oxygen and the hydrogen, and there's a covalent bond between the oxygen and the other carbons. I got it. It's a covalent bond. So the answer here was carbon, hydrogen, oxygen, and they're held together by covalent bonds. So you kind of think through those questions, come up with where you think the answers are. Okay. Second thing, we now have to use the template in the space provided to construct an appropriately labeled graph based on the data. Okay, so looking at this data, I have individuals and I have numbers. That is categorical data. I'm looking at each individual and how their levels specifically were. So this will be a bar graph. Okay, so you'll label your axes. So average glucose level. Make sure you have your unit. So I usually just take whatever's in the title of the chart, and I use that as my title. It kind of helps me to ensure that I've kept my units and I've got everything I need to have. Okay, and then I have my individuals on my x-axis. Okay, so then I would graph them all. Okay, so when I graph them, I would put, of course, they pre-made this side for me, which was great, and they already had all of them labeled, so I already knew those, but I had to label the bottom. Okay, so I put my bars on there. When you're graphing these bar graphs, you want to make sure that you don't shade them in. Number 1 is taking up time. Number 2, it's going to cover up your error bar in a second. So if you do have to shade because you're differentiating 2 different samples, then make sure that you don't cover up the error bar. Okay, and so you may be saying, well, Ms. Jones, what are you talking about with the error bar? So I graphed 170. Here is 170. There's this plus or minus 15. I don't know what to do with that. That is your error bar. You're going to go above by 15 and you're going to go below by 15. Same thing as our second one. We have 190. I'm going to go above by 10 and below by 10. And I would do all of them, but you can follow that right now and you can look at each of these data points and you can see above and below that I went that a certain amount. Okay. Keep in mind, I'm graphing this on computer, so there's a chance that I'm a little bit off, but they should be right. Okay. So I now need to determine one individual who is at risk for developing the disorder. Okay. So these were the people that were at risk. Okay, so they're normal if they are less than 140. Okay, and they're affected if they're over 200. Okay, so I gave myself this little line to kind of help you see where we're looking at. Okay, and so when I look here, and I want to say who is at risk, and they have a significantly different blood glucose level from 4-1. So 4-1 is this person right here. So when I'm saying I want a statistically different, okay, that means that I want the error bars to not overlap. Okay, so if I look here, I can see that this one overlaps and this one overlaps. So 4-2 and 4-4 both overlap. Okay, but then right here, this one doesn't overlap. So I know, oh, I did make a graph. So I can see that this one is completely out. So that would be our 4-3 is going to be the one that is within our range of being at risk, as well as the error bar does not overlap. So it tells me that that one is the one that's statistically significant. Okay. So great job to XYLILES. Great job. I saw that. You got your answer right. Yay. Okay. So there was that one. So going on to 3. C. Based on pedigree, identify all individuals in generation 4 that can pass on the mutation to their children. Okay, so if I'm looking at this generation right here, I need to figure out who can pass it on. Okay, it told me in the prompt that it is a mitochondrial trait because of the fact that I know it's a mitochondrial trait, who is the only person who can pass it on? The only person that's able to pass on a mitochondrial trait is the one that passes on the mitochondria. And the person who passes on the mitochondria is the mother. So I'm looking for the females in this chart. Okay? I'm looking for these circles. Okay, so 1 is able to pass it on because its parent passed it on to it, right? Its mother passed it on. Okay, 2, 3, and 4 also have an affected mother. Okay, 2 is a female and 4 is a female, but 3 is not a female. A male will not pass it on to his offspring because of the fact that he doesn't pass on his mitochondria. Okay. I love that typo. And then here we look at 5 and 6. We have an affected father. The father doesn't pass it on. So 5 and 6 are not affected. So they also cannot pass it on. So we're going to say that 1, 2, and 4 are going to be the ones that are affected. Okay. And they're going to be able to pass it on. Okay. It's not sex linked, it's mitochondrial. It told us in the prompt, which I can't, if I can get back fast enough. But right here, it tells you right here, it's caused by mutation in the mitochondrial DNA. It told you right there, mitochondrial DNA. A lot of us will just overlap that and we don't see that. That's why I was mentioning that we want to underline and circle things that could be important to us. Okay. So knowing 4-1, 4-2, and 4-4, again, it tells us to identify. You probably are okay to just write the numbers like this. But personally, I tell my students to write this as a complete sentence. The individuals are. Like, that's 3 words. The individuals are is 3 words. How hard is it to write 3 words to ensure you get your point? Okay. Okay, so based on the fact that individual 2, 3, I apparently didn't hide the answer key on this one, is affected. So here we're looking at 2, 2 is affected. A student claims that the disorder is inherited in an X-linked recessive pattern. Based on the student's claim, predict which individuals of generation 3 will be affected by the disorder. Okay, so we're saying, here's the claim. This is sex linked. Okay, so ignore all of the things that you were kind of thinking about in terms of mitochondrial. Okay, you now need to think sex linked. Okay, and who passes on sex linked? Anybody who's affected that has an X. Okay, that has the affected X. Okay, so when we look at this, we've got this affected female right here that passes it on. Okay, so she can pass it on to the 2. She can also pass it on to 4. She can pass it on to 6, and she passes it on to 8. Okay, but we were trying to say who's going to be affected. Okay, well, a male, they get one of these X chromosomes, right? And so we are showing that it can't be 2 or 6 because of the fact that they didn't get that second X from their father. Their father does not have an affected X, and thus we still see that they were affected. 2 and 6 should not be affected if it was sex linked. And so we got to, of course, say that they're out of the whole thing. Okay. So of course we were looking at 3, 4 and 3, 8 would be the individuals that would still be affected by the disorder. And our justification is talking about the fact that we wouldn't see it in 2 or 6. Okay. I'm so sorry that I ran long, y'all. I'm just trying to make sure I give you as much as I can give you. So unit 7 is on evolution. Okay, this is where we see natural selection. Okay, this was, do not say survival of the fittest. This is where we see the descent with modification or natural selection. So this, in this population that's on the dark rock, we have these mice, and I'm assuming you've done the rock pocket mouse. Shout out to HHMI with that rock pocket lab. I love it. These Snickers of the desert, of course. And so if you're looking at this, these mice that are darker are able to kind of blend in with the environment. And so because of the fact that they can blend in with the environment, these kind of lighter mice do not. And so they are consumed by the hawks and these other visual predators that can kind of eat them and go, rah, eat them. Yeah, rah, Tala. So they eat them and they get rid of them. And so then we see that our population over time is becoming more of this darker complexion and this darker color. Okay, so that's that natural selection. Okay, we got to make sure we mention things when we say natural selection. Okay, and we're going to see that an individual is more likely to survive, and that the individual is more likely to pass on those traits to its offspring, okay? And its offspring are more likely to survive. It's going to have more offspring, okay? Be very careful you do not talk about individuals are going to evolve. A population evolves. This population of mice evolved so there was a more of the individuals that were darker. There was an increase in the allele frequency in the population, okay? Do not say that there's like anything, like the only thing that causes this is a mutation that gets passed on. So it has to be in the gametes. So if you have purple hair, your children are not going to have purple hair because it's not in your gametes. Well, I guess it could be in your gametes, but most of y'all with purple hair don't have it in your gametes, okay? So on to Hardy-Weinberg, okay? So if you've never watched the Paul Anderson video of the TED Ed, the 5 fingers of evolution, just literally Google 5 fingers of evolution, okay? Then you're going to see that there are this way to kind of help remember. So our little finger is meant to kind of help us to remember that we've got, we need to have an extremely large population size. Extremely large population size is going to allow for genetic drift to not have an effect, okay? The second one we have, of course, is that we want there to be random mating. If it is sexual selection anyway, there are certain traits that are more favorable. Because those traits are more favorable, it's going to lead to evolution, okay? We want there to be no mutations. We can't have any new alleles added to that population, okay? No new ones. And our finger is for gene flow. We want there to be no gene flow. Nothing can enter, nothing can leave my population. And then, of course, my thumb, no natural selection. If any of these 5 things are violated, then we could see that the population could evolve, okay? And when I say evolving, I'm talking about Hardy-Weinberg changing, right? I'm talking about the alleles changing, okay? If my allele frequency changes, if my genotype frequency changes, if any of those numbers change, the population is evolving, okay? You've violated one of these 5 rules, okay? So we will do a math in a little bit. Do not worry. I have a fun math question for you. So the other thing we have is phylogeny, okay? So there's probably going to be a phylogenetic tree, whether you're constructing one or picking one out of a multiple choice. There is a phylogenetic tree somewhere on your graph. I feel it in my bones. So this is a cladogram. They gave you a character table, and from that character table, they asked you to figure out which one was more accurate. This is due to morphological characteristics. And then they're going to ask you, okay, now this was from 2023, okay? They said, we need you to put on the trait for extra tooth material. So the way that I always think about this is like you're walking down a road, you're driving down a street, any of these traits that you pass by, you pick up or drop off, okay? So if you need to have the trait for the bovine, that means that on this path right here, I need to somewhere get that extra tooth, okay? So which one does the giraffe have the tooth? Nope. So that means it had to have happened on the pathway that went just to the bovidine, okay? And the same thing here with the whatever, the cypre, whatever it is, it's also on that. So they have independently gotten this trait, okay? So you can actually have 2 individuals get a similar trait due to convergent evolution. This is convergent evolution, similar environment, even though they don't have a direct common ancestor, okay? We also could see phylogenetic trees. Phylogenetic trees are gonna look like this. They kind of look like a tree. And hey, this works out for your question you just asked in the chat. And then, of course, this is a cladogram. Okay, so on this, this shows us the different bears, and we're looking at a timeline. So they're wanting to know, like, you know, how long ago things happened. So something I want you to make sure you notice about this is that there's this branch point. So right where I'm putting that little dot, that's a branch point. Okay, that means that's the common ancestor of both the European brown bear and the polar bear. Okay, this was arbitrarily placed. There was no reason why the European brown bear was on the top and the polar bear was at the bottom. So you can rotate at any time that you have one of those branch points. So we could actually have had polar bear here and European brown bear down here. So you're able to rotate them. Okay, versus we also could have a branch point here. Okay, so that shows us that the Asian brown bear and the Western North American brown bear could also rotate. Okay, they could give you an amino acid chart or an nucleotide chart, and you have to then figure out where you're going to put them. Okay, if you have to make a phylogenetic tree, they're going to give you a template, and you're going to fill it in. It'll be like question 5 on the AP on the FRQ. I don't think it will be, but it could be. So here we're looking at where are the most similar differences. Okay, so the black bear and the brown bear, they only share one difference. Oh, I'm ahead of myself. I'm sorry. So on the phylogenetic tree, they asked us when was the most recent ancestor of all the brown bears, and you would have said like 3.4 hundreds of thousands of years. Watch the axes on your questions. Sometimes they will flip it on you, and so this is going 1 to 50, and so a lot of students got this wrong because they like quickly looked at their axes. Okay, so make sure you read the axes correctly. Anyway, so the black bear and the brown bear have one amino acid difference here. Okay, so because of that difference, we know that they're going to be placed together on that branch point. So we have black and brown. We then have our polar bear because it has 7 and 8 differences from the black and brown bear versus the panda bear has 33, 34, and 40 differences between the other bears. So we know that the panda bear would be my outgroup. And y'all know that the panda bear is actually more related to a raccoon than it is related to other bears, or at least I hope you know that. Okay. We also have a branch point, so the black and the brown bear could rotate here. I arbitrarily placed black and brown. Okay. Whew. So did I answer everything? Yeah. Okay. Oh, and then last but not least, evidence of evolution. So there's a lot of different evidence of evolution. So you could talk about like homologous structures. Homologous structures mean that they have the same structure because of the fact that they had a common ancestor, and that common ancestor, of course, has that trait that we then pass on to our offspring. Okay. You could also talk about like embryology. They have similar embryonic development. You were born with a tail. You were in embryonic development. You had a tail because our ancestors had a tail. You could also talk about like looking at molecular evidence, so the DNA or the proteins. And so DNA is going to be more accurate than protein because you could have a silent mutation, and the silent mutation could then lead to a difference in the DNA that doesn't show up in the protein. So DNA and protein are more accurate, and DNA is more accurate than the protein. Okay, and then of course you could use morphological data like they did in that this last phylogenetic. This example right here, they use morphological data to make the phylogenetic tree. I'm sorry, the cladogram. So 5 new species of bacteria were discovered in Arctic ice core samples. The nucleotide-base sequences of the rRNA substances were determined for the new species. The table below shows the number of nucleotide differences between the species. So this is kind of what we might see on the exam in terms of a phylogenetic tree question or a cladogram question. They might ask you to pick out which one it is. And so we look here and we can see that the number 3 and 4 have one difference. So I know that 3 and 4 have to be kind of branched together, okay? And so we know that that means that this one is out because 3 and 4 are not together on a branch point. And then over here, I can see also that 3 and 4 are not together on a branch point. So I can cancel that out. Versus here, I see 3 and 4 together and 3 and 4 together. So I know that those are 2 valid answers potentially, okay? Then I look here and I say, okay, well, 1 and 2 have 3 nucleotide differences. So 1 and 2 need to be branched together. Here I see 2, here I see 1, that one's not it. But here I see 2 and 1 branched together. And of course, 5 has the most differences from all of them. So I know that they're going to separate out. Analogous structures are due to convergent evolution. Analogous structures mean that they look similar even though they're not similar. So a flying squirrel and a sugar glider would be an analogous structure. They both have that little structure underneath their arms that allows them to jump out of a tree and kind of glide. And so because of that difference, we're going to see that they're actually, one's a placental, one's a marsupial, and because of that difference, we know they're not actually related to each other, but they have similar structures because they were in a similar environment. That similar environment then leads to them, of course, having a similar trait. And I see some of the answers coming in the chat now. So great job to Sarah and Rid one for the first answers on saying that it was C. Great job, y'all. Okay, so here we have a numeric response. There is no numeric response on your test. They could be found in a multiple choice section, or they could be found in a free response question, okay? But you don't have an official numeric response. But I did want to talk about some math real fast, okay? So scientists are studying a population of flowers with 3 different color phenotypes. The color phenotype demonstrates incomplete dominance. The scientists collected data on the color of 100 flowers. The data shown below to test the hypothesis that the population is in Hardy-Weinberg. The scientists performed a chi-square test. Yes, we're going to do a chi-square. I know you're so excited to do chi-square. My students are so tired of chi-square, but you're excited. I know you're excited. So they gave us the number observed. We had 70 that were red, 20 that were purple, and 10 that were blue, okay? And so when we do this test, we, of course, need to figure out, well, what am I doing? Well, Hardy-Weinberg had this formula. It was p squared plus 2p cubed 2 plus q squared equals 1, then p plus 1 equals q, okay? So I need to verify, is this what's happening? But the problem is that I need to count my alleles because one of the values is going to come from my p squared, 2p cubed, q squared, and then one of my values is going to come from what I visibly see, right? So I need to do the counting alleles method, okay? So to count my alleles, I'm going to solve for my p, okay? I'm going to say 2 times 70. Why am I doing 2 times 70? Because there are 2 of these capital A's here. And I'm going to add 20 because there's 1 capital A. So that will tell me that my P is 0.8 because 160. So 2 times 70 was 140 plus 20 gave me 160. And there's 200 total alleles because there's 100 individuals. They each have 2 alleles. So that's 200. So 160 divided by 200 gives me 0.8. I would do the same thing for my Q, okay, where I've got 10 individuals that are homozygous recessive. I do 2 times my homozygous recessive because they have 2 of those alleles plus my heterozygous because they have 1 of those alleles, giving me 40. And then the 40 divided by the 200 gives me 0.2. Okay, so now that I know my P and my Q, I can use it to solve P squared, 2PQ, and Q squared. So to find P squared, I take my whatever my P is and I square it. And that gives me 0.64. So that tells me there should be 64 individuals because there was 100 individuals total. So that tells me there should be 64 individuals that should be this homozygous dominant red color, right? And then this is a typo. That should be 2 lowercase a's. I'm sorry, y'all. So in this one, I'm going to do my Q squared. So Q was 0.2, 2 squared is 0.04. Do not write 0.4. Be very careful that you do not mess up when squaring these decimals. They don't act like you think they do. So then 0.04 times 100 gives me 4 individuals. So that tells me there should be 4 that are blue. And again, I'm sorry for that typo. That should be 2 lowercase a's right here. Okay. And then we solve 2pq. So 2 times my p being 0.8 times my q being 0.2, which gives me 0.32. So that tells me I should have 32 individuals. So when I do my chi-square, oh yes, I use this beautiful table. I always use a table. I don't know why. It just makes sense for my brain to kind of conceptualize it and put it into little places like this. So I took my observed. I had 70, I had 20, and I had 10. Okay. So that adds it to 100. Okay. It would not be a 9 to 3 to 3 to 1 ratio because of the fact that we're not looking at a heterozygous cross, a dihybrid cross, actually, and we're looking at incomplete dominance. So here we have our expected. This is what we solved using the counting alleles method, right? We counted the alleles, we figured out our P, we figured out our Q, and then we did that with the Hardy-Weinberg, which is an estimation, okay? Since we know the number of alleles, we have to always use the counting alleles method. So it's an estimation, okay? So we then do 70 minus 64, giving me 6, 20 minus 32, which should be negative 12, sorry for that typo, and 10 minus 4, which gives me 6. I then square it, give me 36. 144 and 36, which gets rid of my negative, so it's perfectly fine. And then we go and take our 36 divided by our expected. So observed minus expected squared divided by expected will give me 0.5625, 4.59, which then added together gives me 14.06. Am I done? Oh no, I need to figure out if it actually affects it, okay? So here I have this degree of freedom chi-square, okay? No, this is not looking at a heterozygous thing. This was looking at a incomplete dominance, and they told you in the question, okay? So on this, okay, and this is where students get tripped up, and this is why I'm putting this in this review, right? They were asking about Hardy-Weinberg. Hardy-Weinberg is looking at a P and a Q. There are 2 alleles, okay? Which means my degree of freedom is 2 minus 1, which is 3.84, okay? So that means I'm going to compare this to that. Okay, so 3.84 is less than my calculated value. So if what you find is greater than the value in the chart, you're going to reject your null hypothesis. This population is not in Hardy-Weinberg. Okay, if the value you solve is less than the value here, you're going to fail to reject your null. You can't accept your null, but you can fail to reject your null. Okay, so great job to everybody who just got that one right. Okay, and on to Unit 8, because Miss Jones is still talking for some unknown reason. This should say ecology. I'm sorry I messed up here. Clearly, I was at the end of trying to get this PowerPoint put together, and I was tired. So we have our energy flow. So in energy flow, we're looking at that there's about 10% that goes from one energy level to the next energy level. Okay, each time you're losing heat. So we have our producers. We have our primary consumer, secondary consumers, tertiary consumers, quaternary consumer. Primary consumer eats, of course, our primary producers. Secondary eats our primary. Tertiary eats our secondary, and so on and so forth. Okay, and so what we see is that they can ask us questions about if something happens to one of these levels, okay? So if there is some reduction in our primary producer, okay, then we're gonna see there's gonna be a decrease in the whole level up, okay? If there's a decrease in my tertiary consumer, that means my secondary consumers are gonna increase because of the fact that they're not being consumed as quickly, okay? And so you're kind of figuring out that, of course, we've got this trait, right? We also have population ecology. So this is where we see some more maths, right? So here we're seeing that we've got exponential growth. So this is our J-shaped curve. So there is unlimited resources. There's nothing to keep that population in check, okay? And then we have, of course, our S-shaped curve, okay? This is where we see our population size kind of having a carrying capacity, okay? So once we reach that carrying capacity, we're gonna see that it kind of levels off because of the fact that there's something that's limiting that population, okay? And so that would be our logistic growth. So you may have to do this math where you have your R max times your N. That's going to be showing you your change in your population over time. And if we're in logistic growth, which means we have our carrying capacity, that K stands for carrying capacity, we'll do our R times our N, and then we do K minus N over K. So carrying capacity minus our population size over the carrying capacity. The closer that your population size gets to carrying capacity, the decreasing of the rate. The rate is just looking at the slope. So if they ever ask you for a rate on the AP exam, they're asking for the slope of the whole thing, okay? So as you see, our slope decreases because our rate is going to decrease as we approach that carrying capacity, okay? And then last but not least, we have our community ecology. Community ecology is just looking at the interactions that the different individuals are having in our community, okay? So we have, of course, predation. It's going to be positive for the predator, negative for the prey because they're being eaten. Mutualism, they can both be benefiting. And so this is actually an obligate mutualism because of the fact that they have to live together. So like the termite and the protozoa, they have to be together in order to survive. I don't know if this one is. I thought this was termite when I first looked at it. I'm sorry. We also have competitions. So they could be competing for the same resource. If they're going through competitive exclusion, that means one of them is not going to survive, so we'll pass out. I have the calculator shortcut for chi-square on my chi-square video, by the way. We also have parasitism. So we have a parasite. It could be an endoparasite or a ectoparasite. An endoparasite means that it's going to be on the inside, ecto is on the outside, like a flea is an ectoparasite versus like a tapeworm is an endoparasite, okay? And then we have commensalism where one of them is benefit and one of them has no effect, okay? So we have these beaked whales. They feed at various depths. They defecate at the ocean surface. Nutrient-rich whale feces deposit the surface water, supply nutrients for algae that are eaten by surface-dwelling fish. Okay, so there's these fish, and they eat the nutrients from the feces that are full of nitrogen. Okay, so which of them best predicts what's going to happen? Okay, so here I have a whale, and then here's, of course, the algae that use it, and then here's the fish that eat the algae. Okay, so I draw myself a picture. It kind of helps you to figure out like what you're seeing here. Okay. And so they say, what would happen if there is a decrease in the whale population? So there's no more whale, bye-bye whale, okay? What happens, okay? So if there's a decrease of the whale, that means that there's less feces at the top. So it means there's less nitrogen for this algae, which means there's less fish, okay? So there would be a reduction in surface nitrogen concentration, cool, which would cause an algal bloom. We know algal blooms are not due to nitrogen being in absence. We know that there needs to be a large amount of nitrogen. That's what causes the algal bloom. Surface fish population would decline due to reduced population of algae. So the algae doesn't have enough food, so we would see less algae, and since we have less algae, we have less fish. That's logical. And then the surface remaining whales would accumulate mutations. That has nothing to do with this. The remaining whales would be forced to forage in deeper parts of the ocean. Again, this has nothing to do with the whale being gone, okay? And so that was our answer. So testosterone oxyreductase is a liver enzyme that regulates the testosterone levels in an alligator. One study compares the testosterone oxyreductase activity between a male and female alligators. There's a pristine Woodruff versus a contaminated Acapulco. And the graph above shows the findings, okay? So they want to know what of the data in the graph best supports which of the following. Okay, so we're looking at this graph and we say, okay, well, this one's pristine and this one's contaminated. You can ignore my children talking in the background. I'm so sorry, y'all. And so we want to see, okay, how is the male affected and how is the female affected? So here I see the female is going to have a decrease in its activity because of the fact that it's between the pristine and the contaminated. So the contamination must be decreasing the amount of reductase usage, okay? Versus if we look at the male, these error bars are overlapping. So since the error bars overlap like this tells me that this data is not statistically significant. So the contamination is not having any effect on the male's oxyreductase activity. So if we look through our answer choices, environmental contamination elevates total activity in females. That is false because we see it going down. Environmental contamination reduces the total testosterone in females. That's true. We do see that, okay? And then we say elevating in the males. It does not elevate the males. It doesn't reduce. You could look at the bars and say that it does reduce, like if the mean does, but the error bars the error bars do overlap, so it tells us that D is wrong. And I'm seeing a whole bunch of B's in the chat, so great job to everybody, okay? So that's all of the units. But let's think about like science practices and kind of what happens here. So we have independent variables and dependent variables. In your FRQs, you're going to be asked about these variables, okay? So independent variable is going to be what the scientist is changing. What did they modify? And because of the fact that they modified it, then what happens because of that, which would be your dependent variable, okay? So in the question, it's going to say to you, the researcher changed da 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 da. That's your independent variable. The researcher measured da 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 da, or it's going to show you in the data table that measurement, okay? So independent is what the researcher changed. The dependent depends on that independent, and that's what's going to get changed. That's what they're going to measure. That's what happened in the experiment, okay? Your null hypothesis is going to be that the independent variable has no effect on dependent variable, okay? There should be no difference between your control and your treatment group. They should be the same, okay? You can't say like that there's just nothing there. You have to just say that the two are the same, okay? So in your null hypothesis, independent variable has no effect on dependent variable. And be very specific about the question, okay? Like they give you the prompt, be very, like, use your prompt. Tell them back to them. If you just say independent variable and dependent variable and don't identify the independent and dependent, you're not going to be able to get points on that. You have to be very clear about your question, okay? And then positive and negative controls. Your positive control is one that you know what is going to happen, okay? Versus your negative control is going to be like without the treatment group. Okay, so in this experiment, this is our transformation lab. Okay, you had your minus P low. That means it doesn't have the plasmid. Okay, so it's not going to be able to glow and it's not going to be able to grow in the presence of antibiotics. Okay, we have an LB plate here. So this makes sure that my bacteria survives, that it is actually viable bacteria, that it is good to go. Okay. And then over here, so that would be my negative control because it doesn't have the plasmid, okay? And because it doesn't have the antibiotic. And then here, I don't have the plasmid, okay, cool. But I do have the antibiotic. So this is, I would expect that since it doesn't have plasmid, it does have antibiotic, that it's not going to survive. And so this ensures that when I do look at this lab that does have the plasmid, that allows me to attest that the plasmid is the reason why it grew. So this right here is my positive control. So the first one's my negative control, second one's my positive control, okay? So other things about your graph, make sure you know what types of graph it is, okay? If you have categorical data, that's a bar graph. If you have linear data, so you have time at 5, 10, 15, 20, you have concentrations, 0.2, 0.4, 0.6, 0.8, that is a line graph, okay? If you have 3 sets of data and you're comparing 2 of them and then you've got just like individuals or locations or something like that, but you're comparing these 2 sets of data and then you got this random third one, that's a scatter plot. And you could have a box and whisker. We have not had one where you've had to graph a box and whisker, okay? But there is this thing called a box and whisker, and it was on question 6 last year. So if you want to like look over a FRQ that has to do with a bar and whisker and make sure that you understand how to do bar and whiskers, okay? That's what this is, okay? So this is a bar and whisker. I'm sorry, not bar and whisker. Y'all, I'm sorry, I'm tired. Box and whisker, box, it's a box, see, box, and then you got these little whiskers. Anyways, so this is showing us a, like if this was all of your data, okay, then you're going to say, okay, from here to here, this is 25% of your data. From the top of your box to this little line in the middle of your box, that's going to be the next 25% of your data. From the line to the bottom of the box, that's the next 25%. And then the line to the bottom is the rest of the data, right? It's the very last 25%, okay? And so we're going to see that this is all our structured data, okay? And then the line in the middle is your median. So last year they asked the students to explain which one had the lowest median, which one had the highest median. And all you had to do was just be able to look at the box and whisker and say, oh, this one had the lowest number. Okay, so it wasn't really a hardcore question. They just wanted you to look at it. And then they also were asking about the stretch of it. I did an FRQ Friday video on this. So it's, I think I don't know, maybe like 29 or 28. But you can see it says 2023, number 6 on it. It's on my YouTube page. So things about your graph. Number 1, you want to label your axes. You get points for labeling the axes and scaling correctly, okay? Scaling your axes, this is important, okay? Each box has to be the same amount. Do I have to label every box? No, I do not have to label every box, but each box has to still be equal. So if you're counting by 5s, okay, and you go up every 2, then you're just gonna write 10, 20, 30, 40, okay? If you're counting by 2s, then and you want to only write on every 5th box, okay, you would do 5 boxes, there's 10, 5 boxes, there's 20, okay? Make sure that each box is the same amount, okay? Graph your data points, making sure that you do correctly graph them, and then make sure you have your error bars. Most recently, we've seen you get a point for your axes, a point for the correct graph, and a point for your data and your error bars. In previous years, we've had a point for axes, data points, and error bars. So there's usually those kind of mixes and mingles, and then they're going to ask you to analyze it. Okay. So more math that's not on your formula sheet. So identify the percent, I'm sorry, calculate the percent change in APase activity in the wild type yeast strains in the high PI compared to the low PI. You don't really need to know what the question's asking about. I just wanted you to like pull this two data here, okay? So they've been asking for percent change the past 3 years. It's been on the exam asking percent change, okay? So with your percent change, what we're going to do is we'll do our final value minus our initial value over our initial times 100. Okay, so our final value was 17.3. Our initial value was 0.5. So we did 17.3 minus 0.5 over 0.5 and multiply by 100. That gave me 16.3 over 0.5. That gives me 33.6 times 100, and that gives me 3,360%. Okay, because it says to calculate the percent change. So you need to move this back into like percent format. Okay, I would not use an erasable pen on the exam. It does go away. Okay, so I think at this point, since I'm almost at 2 hours, I should not do a Q&A. But on my Instagram, if you do not already follow me, on my Instagram every single day, so Sunday through probably Tuesday, I'm going to have a question box, and you can ask whatever questions you have on that question box, and I will answer them in my story. Been doing this since February 1st, and so all of the questions that have been asked since the beginning of my review are all stored in my highlights. So you'll see them at the top of Instagram little highlights. It just says Q&A 1, 2, 3, 4, whatever. I've got TikToks. You don't need to follow the TikTok. It's fine, whatever. And then of course you're already on my YouTube panel because you already know that. Okay. I am going to go live with Marco Learning on Wednesday. So if you want to see something again, I'm probably going to have to cut it a lot for my Marco session because I have to stay at an hour because I'm on his YouTube page. So yeah. There's that. So I hope that this was helpful. I'm sorry that I ran over by like 53 minutes. But I hope that you remember that you are penguins, and you are dressed for success, and you're going to rock this whole test. And I do know that there is a weird lag, and so I don't want all of that message to go away. So I'm here for you. I'm excited for you. I'm waiting for this video to catch up. So I'm just going to look at what's in the chat. Yes, it's going to be 8 p.m. Eastern time with Marco Learning. Thank you for being here and for supporting this. Let your friends know AP Bio Penguins that take AP Bio next year, because I'm, happy to help y'all however I can help. And of course, shout out to Miss McClinton. I cannot talk right now, y'all, because she has been like killing it in the chat, helping you all with all of your questions. And I'm sorry that I wasn't able to answer them as fast as I wanted to because there was 200 of y'all in here. I'm so proud of y'all. Anyway, so I think I have killed enough time, and I hope